Assalamu alaikum and good afternoon to our esteemed guests and delegates. A very warm welcome to the afternoon sessions of the conference. I am Dr. Shamir P.S.,
I'm Dr. Sandhya.
Junior Residence in the Department of Pharmacology, JNMC AMU. This session will feature oral paper presentation for the METEX prizes. We are honored to welcome Professor Azhar Khan and Professor Abul Kalam Nejmi as the chairpersons for this session. Professor Azhar Khan, a professor at the Faculty of Applied Sciences and Biotechnology, Shulini University, received his PhD in biochemistry from JNMC AMU. He pursued postdoctoral research in the field of diabetes and obesity-mediated hypertension from the University of Houston, Texas, USA, and Auburn University, Alabama, USA. Dr. Khan received the New Investigator Travel Award Trainees for HBPR 2012 by the American Heart Association, USA.
Professor Abul Kalam Nazmi, Head of Department of Pharmacology at SPER Jamia Hamdard, New Delhi, has been awarded many national and international honors, awards, and fellowships, including the Career Award for Young Teachers by AICTE India and the Uppsala Best Paper Award at the Annual Conference 2009 of the Society of Pharmacovigilance. He has also received several awards for publications in high impact journals and international travel grants. Now, let's proceed with the presentations.
Good morning, good afternoon, everyone. So, the first speaker that I would like to invite is Dr. Maharajan K. He is Assistant Professor, Department of Zoology, University of Hyderabad, Prayagraj. So, the dice is over to you.
Thank you, sir. Good afternoon to everyone. So today I am here to present my part of my PhD work, which is carried out in DRDO Center at Bharatiya University, Coimbatore, on developmental toxicity of pyriproxyfen in zebrafish embryos. So currently I am an assistant professor at University of Allahabad. So coming to introduction, pyriproxyfen is a pyridine-based pesticide widely used against agricultural field as well as in mosquito control mosquito population. It mimics the juvenile hormone and inhibits the embryogenesis of insects. So in India, it has been used as well as in agriculture and in pest control against Culex and Anopheles mosquito. Recently in Brazil, this pyriproxyfen was suspected for the microcephaly in newborns through like the mother by drinking the water sources. So the WHO guideline for the use of pyriproxyfen drinking water sources at the maximum final concentration of 0.01 mg per liter. Yeah, so recently Belanguer et al. Reported the presence of pyriproxyfen in environmental sources in Juga River, Spain at 89.66 nanogram per liter. The exposure of pyriproxyfen in aquatic ecosystem can risk the aquatic organisms. It can cause mortality in non-target organisms like daphne at the range 10 microgram per liter, as well as in honeybees, crab, shrimp, and daphnia. It causes the toxicity. It also causes the endocrine disruption in daphnia, zebrafish, and as well as in mice. However, in case of fish, it is the main organism present in the aquatic water body. The studies are limited to lethal concentration determination, and the adverse effect of pyriproxyfen in aquatic organism is very rare, scarcely reported. Hence, the aim of this study was to evaluate the developmental toxicity of pyriproxyfen using zebrafish model. So as you see here, like it has a lot of impact in other aquatic invertebrates like Daphnia and crab, but the higher order vertebrates in fish model, it is lacking. So as we know, zebrafish is a well-established alternate animal model in the toxicology as well as in biomedical research. So it shows genetic similarity, genetic as well as physiological similarity with human. And this fish model is considered under a 3R principle up to 120-hour post-fertilization. So it also offers many technical advantages like optical transparency and amenability to genetic and chemical screens. And this is like showing the whole life stages of zebrafish from embryo to larva, juvenile, and adult at different time period, 0 3 day, approximate 3 day, it will hatch out into larva and then to juvenile and adult stages. So the main objective of the study are to study the adverse effect of pyriproxyfen on early embryonic stages, as well as to study the impact on oxidative stress and biochemical markers, to study the antioxidant responses, and to evaluate the neurotoxic and genotoxic potential of pyriproxyfen in zebrafish embryos. So coming to the experimental design, so the fish were maintained in standard laboratory condition using zebrafish growth chamber, and the spawning was set and the embryo was collected, and it was maintained in the embryo medium. So the 3 concentration was chosen like 1.16, 0.33, 1.66 microgram per ml based on the effective concentration EC50, and 50 embryos were exposed in 50 ml embryo in 100 mm dish. So it is conducted based on the embryo toxicity, fish embryo toxicity test OECD 236, and the exposure was given at 3 HPF hours post fertilization, and every 24 hours the medium was renewed, and the endpoints like malformation, heart rate was determined from 24 to 96 HPF. So the nominal concentration of this exposure concentration was determined at during exposure and after 24 hours using GCMS analysis, and the various endpoints like heart rate measurement, acetylcholine esterase staining, genotoxicity assay like Comet, agridine orange staining, again acetylcholine esterase, enzymatic determination, as well as various biochemical oxidative stress and antioxidant responses were measured at 96 HPF. So these are the complete methodology used for this study, like for developmental malformation through inverted microscope, heart rate measurement as well, and biochemical oxidative stress and antioxidant responses through Hariva spectrophlorometer, and all these parameters were measured through microplate reader, and again genotoxicity assay through inverted microfluorescent microscope. So this is about statistical analysis. Graphite prism was used, image was used to measure the heart size and olive tail moment in Comet assay, and the results were expressed in mean plus or minus standard error mean. So coming to the results, so this is about, this slide shows the measurement of nominal concentration in exposure medium. This is an nominal concentration. This is a measured concentration at T0, 0 hour and 24 hour. There is a deviation about 25%, about like 30% deviation between 0 hour and 24 hour concentration. So the renewal, semistatic renewal of this pyriproxyfen is to make sure the concentration, exposure concentration is the same throughout the period. So this slide shows the developmental toxicity, like developmental deformities, and the first one panel shows the control development, normal development of the zebrafish embryo at different time point, and 0.16 there is minimal or no deformities at lower concentration. However, in higher concentration, we can observe some of the deformities like pericardial edema, scoliosis, yolk sac edema, and delayed hatching, as well as in the higher exposure concentration, particularly at 72 and 96 HPF. So this slide shows the effect of pyriproxyfen on heart development. The first row shows the control development, and this is highest exposure concentration. And we can see the heart development, the pericardial edema, and it leads to the stretching of heart muscles. And the heart rate is increased, significantly increased at 48 and 72 HPF. However, it is decreased in the 96 HPF. This figure shows the heart length measurement at the zebrafish embryos in 1.66 microgram per ml concentration. So it is significantly increased. The stretching of heart muscles is significantly increased, and reduced heart rate was found in the 96 SPF. And this is about oxidative stress markers like ROS, reactive oxygen species, lipid peroxidation, and biochemicals like LDH, lactate dehydrogenase, NO, nitric oxide, and AP activity. So there is a significant increase in oxidative stress and as well as in biochemical markers, particularly NO. And antioxidant responses like SOD, superoxide dismutase, catalase, and GST, glutathione S transferase, peroxidase, as well as glutathione S reduced glutathione. So there was significant increase in this oxidative stress antioxidant markers, as well as in reduced glutathione, the level was significantly decreased. So coming to neurotoxic effect, I have measured through acetylcholine express inhibition through staining as well as enzymatic measurement. So the dark brown color indicates the intensity or activity of acetylcholine estrase. The intensity, if it fades, the activity is decreasing. So it is clear at 96, at 48, the at higher concentration it is decreasing the acetylcholine, which means the inhibition of acetylcholine activity. This is this image shows the enzymatic activity, which also shows the similar effect. And this slide shows the genotoxic effect of pyriproxyfen using by Comet assay. Control 0.16, 0.33, and 1.66 microgram exposure. So again, in this slide also, like results also, higher concentration showed the significant DNA damage, which is also measured by Ollit whale moment. We can see the significant damage in the 1.66 microgram per ml exposure. Again, agridine orange staining, which determines the apoptosis in the live embryo. So the green fluorescent indicates the apoptosis cells in the developing embryos. So this in control, the means the larva is not clear here because it is a dark background. So it is, the larva is in this axis. In control, again lower concentration 0.16, 0.33, and 1.66. So in 1.66 we can see like significant apoptosis in the mainly in the head region. This reveals the again the genotoxicity marker. So overall, so after pyriproxyfen exposure, like it causes the oxidative stress markers like ROS and LPO, and it increased the antioxidant responses. It caused the genotoxicity like in Comet assay as well as in agridine staining. So I take this opportunity to thank my guide, Dr. M. Ramesh, Professor and Head, Retired, Department of Zoology, Bharatiya University, as well as Dr. P. L. Rao, Director, Dr. K. Kadirvelu, Scientist, DRDO, Bharatiya University Center, Dr. Babu Rajendran from Bharatdas University for GCMS facility, Dr. K. P. Singh, Professor and Head from University of Allahabad. And I also thank the Organizing Secretary, Dr. Rahman Sir, for this opportunity to present my work here. Thank you very much.
Okay, was nice presentation and nice finding. I have just one question. Is there anyone from the audience who want to put forward any question? Yeah.
Yes, sir. Yes, sir. So main action is induction of oxidative stress, because oxidative stress leads to the various changes like causes, particularly genotoxicity and everything. So oxidative stress is the main responses I have found. Yes, sir. Yeah. Which one? Yeah. So third one, yes, LDH, lactate dehydrogenase. Yeah, there is some alteration which we need to find in different, maybe different method or something. We need to understand why it is decreasing at a higher concentration. Yeah, yeah. Usually, yeah, I understand that marker should be increased at a higher exposure concentration. Yes, I agree. No, after that, like no, we stop with this. We need to perform additional experiment to find why this is happening. No, man, we couldn't means justify why it is decreasing in particularly at higher concentration. That's what like in every other aspect of parameters, it shows the, like significant increase at the higher concentration, but only in this parameter it is decreasing in the metabolic activity particularly. Yeah.
I just have one question to ask. Since you talked about the problem created by this compound, so is there any solution so that we can get away from this compound causing a toxic effect to these aquatic life? So is there any solution to it?
Sir, solution usually it is always said that like use of biopesticide or like environmental friendly pesticide, but because here we use this pesticide in for the different aspect, like mainly in one thing in pesticide in agriculture field as well as in like public health concern to control the mosquito population. So yeah, definitely we need to find alternative when we consider this kind of chemical pesticides.
Yeah, so but even then we can take an alternate approach. Like for example, we can look into the bioremediation effect, how we can bioremediate these compounds from in these aquatic sources,
Yeah.
so that we can
we need to like find the solution, like how we can like after application like if we see the effect desired effect, then we have to make sure the compound is not present in the environment.
Exactly, because ultimate goal is to protect the environment and to get away from this.
Yeah.
So we always talk about the problems. This is a problem that has been created.
Definitely, yes. We have to look
What
for
is
the solution. Yes, of course. So
Thank
first we have to identify the problem of this compound. So this is like initial step. That's what.
Thank you. Thank you.
Thank you. Yes. Yes, it was suspected. Like it was suspected for the cause of microcephaly in Brazil, but the later reports like still research is going on. Like, is there any correlation for the actually the cause of microcephaly was Zika virus, and then it was suspected pyriproxyfen used in the water sources which the pregnant woman taken. Like there was correlation and everything, but still that research is going on. But mostly this was now declared to the Zika virus is the main cause for microcephaly, not pyriproxyfen. Sorry, ma'am, can you? Yeah, neuronal we can relate with the only with the acetylcholine S6 activity, but we need to perform a specific neurotoxicity study. But I have done just general toxicity, not like specific to neurotoxicity. So if we perform neurotoxicity and like based experiment, like behavioral changes and everything, so we can find out what is the effect of pyriproxyfen specific to neuro effect. No, I have not performed in that aspect. Thank you. Sorry, sir. Can you?
Approval of your ethics committee
No, sir.
to perform experiments on
No, as of now in India, there is no ethical concern for this zebrafish embryo. Recently, CPCSA has released the guidelines for use of means for the use of fishes in experiment. For many fish species, we have to get the ethical permission, but not for the embryo. As well as in European countries and US, up to that's what up to 120 hours post fertilization we can use under 3R principle. Like,
what is the objective of your study? Just to see the toxicity of these compounds, or to see also the remedy or solution to these
so initially I need to study because there is no report on this pyriproxyfen on aquatic environment, particularly higher order animals. So I want to study the impact of this pesticide first. So novelty in this case, we can say the results of the compound actually. So this is a study, like most of the toxicity studies are like common for all the compounds, like if it is mainly in agriculture, pesticide or drug screening. These are the parameters we study. So novelty here, I'm the one who first studied about this compound in what is the impact on aquatic environment or toxic means developmental aspects.
Okay, thank you so much.
Thank you.
So now I invite Dr. Barkha Asia from Section of Genetics, Department of Biology, AMU.
So first of all, very warm good afternoon to all of you, and thank you for giving me opportunity to present my oral presentation over here. So today, this is Dr. Barkha Shaki from Department of Zoology, Section of Genetics, AMU, and I'm going to give my oral presentation on Drosophila melanogaster, a promising model to detect neurotoxicity and changes in life cycle parameters when exposed to ericoline. So first of all, let me tell you what is ericoline. Actually, ericoline is an alkaloid which is taken or extracted from erica nut, which is a fruit of erica palm. It is also known as Erica catechu, and it's an oily, odorless liquid. And initially, in our past, people used to take it as a mouth freshener. It has an euphoric effect. It's also been used as a good digestive product, and it is also being used as an anti-bacterial and antifungal. So, because of various reason, it's been used. But here we use Drosophila melanogaster, which is the strain of Oregon R, and obtained from Drosophila Genetic Resource Center, Kyoto Institute of Technology, Kyoto Center, Japan. And we also use this because it has 75% of human disease gene has a recognizable match in the genome of fruit flies. And it has also been used in several experimental setups like to detect neurotoxicity or neurodegenerative disorders like Alzheimer's, Parkinson's, and Huntington's disease, and many more. So here we took Drosophila and we maintained the whole strain in a special diet which was made up of agar, corn, and sugar and yeast, and we maintained the temperature to 25 to 26 degrees Celsius. And on the other side, the compound which we extracted, that is ericoline hydrobromide, we extracted that and we prepared various doses of diet, that is 5, 10, 20, 14, 80 micromolar of diet, which has that ericoline hydrobromide. And we took newly eclosed Drosophila flies, and we allowed those newly emerged flies into that diet, which was having various doses of ericoline. And after 5 days, we prepared a homogenate of the male heads of those flies, and we further performed various experiments like climbing assays, activity pattern, glutathione content, and various lipid peroxidation assays like protein carbonyl content, acetylcholine esterase, monoamine. Also, we try to detect what is the effect on apoptosis, like caspase activities, caspase 9 rice and from apoptotic index. Also, we studied the effect on pupation and emergence of flies. We also studied the what kind of difference is there, or what is the lifespan impact on lifespan, as well as phenotypic analysis on eye. Further, we also here try to study the preliminary test, which is like we take a vial which is of 10.5 centimeter, and we mark that vial up to 8 centimeter. We took near about 10 flights per vial, and we took 3 replicates per group. And we, after acclimatizing those flies, we tap that vial and we try to find out that how many flies are crossing that mark of 8 centimeter in 10 seconds. So with this, we found that as the dose of ericoline is increasing, the activity of flies or the climbing the climbing activities are decreasing. While we also studied the effect on activity through Drosophila activity monitor test. And here you can say there are several vials, like several capillary tubes, and in each capillary tube, on one side there is a diet which contain a particular amount dose of 5, 10, 20, 40, and 80 micromolar of ericoline. On the other side, it's been closed from with the help of cotton swab, and this is in this way it is being aligned, and there is an infrared beam which crosses in between. So whenever a fly moves to and fro, this infrared beam is there is an interruption, and this activity is recorded into a software, that is Actogram J software. And with this, we found that as initially in 5-10 micromolar of ericoline, there was not such changes we found in the activity of Drosophila. But as the dose of ericoline is increasing, we found, look here, that in the highest dose of ericoline, the activity of Drosophila is decreasing in a dose-dependent manner as compared to control. Further, when we studied glutathione content, the glutathione activity, we found that it is decreasing in a dose-dependent manner, while glutathione S transferase, lipid peroxidation, and protein carbonate content were found to be increasing as the dose of ericoline is increasing. Further, the activity of acetylcholine esterase is decreasing, while the monoamine oxidase is increasing. On the other side, we also studied the effect of ericoline on neuronal cells of Drosophila, that is the brain cells of Drosophila. And there are several apoptotic cells, that is, which we can see, these are red apoptotic cells, which were maximum in the highest dose of ericoline as compared to control and various other lower doses of ericoline. So it shows that, yes, it is showing degenerative effect on neurological cells. Further, with this in past study as well, and it is quite evident with this also that caspases is increasing, all those caspases, caspase 9 in caspase 3, and hence the apoptotic index is increasing in a dose-dependent manner. Further, in this pupation study, we also found that the flies which were exposed to higher dose of ericoline, that is this is red color bars, we can see that this is the highest dose, and we found that the pupation rate is decreasing that as the dose of ericoline is increasing. Also, the emergence of flies is also found to be in decreasing fashion. See, these are the highest dose of ericoline. This red bar shows that the emergence rate is also decreasing. Further, the lifespan is also found to be in a decreasing manner. These are the second and third highest doses, that is 40 and 80 micromole of ericoline. They were found that they quite going to impact in decreasing lifespan of Drosophila. Further, we also studied the effect of ericoline on the eye marginal on the eye surface of Drosophila, there is a proper hexagonal shape of the eyes in a Drosophila, which we found that that shape is deteriorated as the dose of ericulin is increasing. So this hexagonal shape in initial doses, there is no further changes, but this one is controlled. And you can see this is clearly evident that how much it is showing the effect in the degradation of the shape, hexagonal shape of the eyes of Drosophila. So with this, we can say that when the newly closed flies were allowed to feed upon the various doses of ericulin, it leads decrease in climbing assay, decrease in activity pattern, glutathione content, and acetylcholinesterase activity. Further, it also results into the decrease in pulsation emergence rate, also lifespan, and also the phenotypic analysis. We can see that the shape of the eyes of blood are deteriorated. And it also leads to the increase in lipid peroxidation of various biochemical acids. And apoptotic index, we found that it helps in the increase in apoptotic activity in Drosophila. So with this, we can say that the possible mechanism of toxicity of ericulin might be results into the metabolic activation of cytochrome oxidase, and it leads to the increase in reactive oxygen species, which leads to the formation of oxidative stress, and further hampering all those biochemical assays inside, and leading to tissue damage, DNA damage, further increases in caspases and apoptosis, resulting to cell death. So with this, we can say that this study not only showed the effect in neurotoxicity, but also changes in the life cycle parameters of Drosophila melanogaster when exposed to ericulin. And yes, it shows that Drosophila proved to be a promising, an amazing alternative model of fire organic. Thank you.
So, questions from the audience, please. Yeah.
Sorry, pardon. Anti-agent. We haven't anti-cancer agent. So that if we go toward cancer, so it has a different pathway and we haven't gone through that. So it is the initial application on healthy cells, not on those cancer cells. Yes, ma'am. Yes, ma'am. We can go through. So it's a good part of further study. Even we haven't gone through it. It is an initial test on this thriller. But yes, it could be studied in a very real manner. If you have thought about cancer, it's a very good part. So we'll go through it. Yes, thank you. Actually, till date, I have already been into Drosophila, but yes, there are various other ways I have studied. Didn't thought about to go ahead, but there are various cell lines as well in Drosophila only. So we are thinking about that to maintain the cell lines in Drosophila transgenic laboratory and to further proceed in the same manner. Thank you. Thank you.
Okay, so I have one question. So, like as ma'am asked, so why it is only as the title is indicating, so you are looking the neurotoxicity. So, would it be good if you took the higher animal model, which could simulate the human physiology in a better way, instead of taking the Drosophila as a model to prove this compound to be?
Actually, sir, there are various reasons that we have chosen this model. First is from safe side to from ethical issues point of view. On the other side, Drosophila have 75% of that similarity with that of human genome. So we are too close to humans. Also, it is easy to maintain, not into a lab, but they are we are into that. They have four chromosomes, like three autosomes and one six chromosomes. As at a single time, in a short duration of time, we can study several progenies of one female lays thousands of eggs, and further their offsprings will lay several thousands of eggs. So from genetic point of view, if we want to study more into it, we are more close to like we are it's giving benefit to study more into a limited time period. So it's very beneficial, not from the point of view that we are saving higher animals from sacrificing, but we are on into it that we are studying more into a limited time. We are saving money, we are saving space, we are saving various other perspectives, and also it's very easy to handle. And we are into that like it's easy to study in a that is known as salivary glands, also known as puff genes, puff chromosomes. So easy to study that eukaryotic perspectives, that replication and translation point of view. So yes, it's a good model to study into the genetic level.
Yes, ma'am.
Yes, ma'am. Yeah. Yes, ma'am. Yes, ma'am. Yes, ma'am. Thank you, ma'am, for the suggestion. Yes. Yes, sir. Yes, sir. Yes, sir. Sir, sir, I have, sir, okay. So actually, sir, haven't I have already so according to my study, we have gone through there. It affects several kinds of like as you said, fibrosis in liver or colon or intestine or any other body part. But if we go towards neurotoxicity, I haven't studied on human neuronal cells. So that's why. Hmm. Okay, okay. Okay. I'm sorry. जी, yes, sir. No, sir.
Actually, what Professor Reis has pointed out is it's very important, pertinent question. I think this recalling or areca catechu product is extensively utilized by general population, especially in rural areas, in pan
Yes.
as a condiment. So a large number of population, large fraction of population is actually consuming this. So have you gone through in literature any report citing all these type of activities or toxicities in humans?
Yes, sir.
Right.
Okay, I got it. So actually, it was reported that there are various symptoms that they have studied, like common people use areca in various forms, that it increases their activity, or it acts as a sedative or somewhere. Various reports gives various results. So there is a confusion whether it is into that progressive way in a in neurological studies, or it is against it. So to confirm what we should do, we have done, we use that product in Drosophila to find out that what are the biological aspects we can go through and take out that which pathway it is hampering and what are the results we are getting.
At the same time, what would you suggest to all those pan eaters in terms of in reference to this toxicity which you have found in your study?
So I will request each and everyone, please पान ना खाइए, aeroplane ना खाइए, सिर्फ आपकी जिंदगी बर्बाद करेगा और कुछ नहीं.
Okay, thank you so much.
So it looks only in singing only. So so we haven't here because we didn't need it in this Vela model.
Okay, thank you so much.
Thank you all of you. Thank you.
So now we have another speaker, Dr. Shehzad Ahmed, Assistant Professor from Department of Medical Limitology and Toxicology, Jamia Hamdard.
Good afternoon, everyone. This is Shehzad Ahmed from Department of Medical Elementology and Toxicology, Jamia Hamdard. And this presentation of mine is some part of my doctoral studies and some part of my postdoctoral studies and some part of my recent studies which is going on. It is based on that, which is telling that the network. It is actually examining that what is the potential of network toxicology to replace or to find out the risk assessment health hazard. We have taken the thalate esters as a model compound. So, what network toxicology is the basic definition, which I would like to highlight here, by Chinese scientist Dr. Zhang, that network toxicology is an important branch of network pharmacology emerges as on the basis of the network biology, how the different proteins, different genes are connected to each other, and what are the effect on means the mutual effect of that. In that I have applied one tool which is a very basic tool and available freely online, that is string database, and it is having the it is telling you the how different proteins are connected to each other. On the basis of that, we can also find that what are the different biological processes, what are the different biological functions which are associated with the with those particular proteins. So if we can approach to a single protein, then we can also see that what are the different other proteins, different other biological functions, different other biological processes, and the different organs which may be associated to the toxicological as a toxicology target of the compound. So for the introduction of my compound, that is phthalate esters, is very well known. That as these are the plasticizers called as they are ubiquitous in nature and found everywhere. And my focus is on the DESP and DVP compound. So the hypothesis of mine was that this is the I was targeting the adrenal gland, and in that adrenal gland, particularly the glucocorticoid biosynthesis pathway, which is found there in adrenal gland, and in this pathway, I was targeting the enzymes which are regulating the biosynthesis of cortisol as the target of the phthalate esters. And if it have this works, then I can see that if the secretion of glucocorticoid is compromised by the expression, either the up regulation or the down regulation of these compounds, then I can see that these many functions may be altered. So first of all, I had done the in vivo studies, in which I took all the endocrine glands, and I was seeing I was seeing that we were examining the toxicity of phthalate esters on the all endocrine glands, that is pituitary, pineal, thyroid, parathyroid, adrenal, and testis, and we found histologically that pituitary gland was affected very much, and sorry, yes, the pituitary gland was badly affected by the exposure of the DEHP and DVP, which is having the different kind of effect. The severe angiectasis was one of the highlighted effect on the pituitary gland, which is telling that the several secretions of the master gland may be compromised if these effects may be there. And this is the micrograph of pineal gland, which is also finding the different effects of these compounds. Sorry, that is the vacuolation of mild pinealoma fibrosis and the infiltration was the highlighted effect on the pineal gland of these compounds. If we see the thyroid gland, it is not. If we see the thyroid gland, then the severe hypertrophy of the follicular cellular loss and the follicular cells were the highlighted effect of the DEHP and DVP on this endocrine gland. And if we see the adrenal gland, then one specific feature, one specific effect of toxic effect, that is means infiltration of the blood into the adrenal gland was a very highlighted effect of the DEHP in the higher concentration. And if we see the testis, these are very famous target of these endocrine disrupting chemicals. The very important target and well-known target, but on the basis of the histological examination, we found that adrenal gland was the second most severe target, second most sensitive target among the endocrine glands after the testis. So adrenal glands may not be ignored in such a way. So I was examining as I told in the starting of my study, that glucocorticoid biosynthesis pathway is one of the targets. So we have taken these proteins of this pathway as for the molecular docking, and we were seeing that if they are having the interaction, what kind of interaction is there by the molecular docking with the phthalate esters, these enzymes. So we have taken these enzymes, the CYP17A1, 19A1, 21A2, and 19A1, and CYP11B2, 3-beta HSD. These are the compounds, these are the enzymes which we have taken for the study, that what kind of effect of these compounds may be there on the particular pathway of that glucocorticoid biosynthesis pathway, which is the main pathway process in the adrenal gland. So what we actually found, what was the indications of the in vivo study? It was reflected in this in silico study, that these phthalate esters were actually inhibiting or interacting in a promising way, with these enzymes. That is these enzymes. If we can see this DNHP, which was having the least docking score, and this is the least docking score by these compounds which you are seeing at the last. These are the compounds, these are the natural ligands or the natural inhibitors of these compounds. So this study is telling that DEHP or DHPH is having the promising effect or the promising interaction in comparison of the known inhibitors. So they may be the good inhibitors of the compound. And this study was also telling that if we see the average glide energy, then this phthalate is telling that is having the least average glide energy, means it is telling that this pathway may be much better than targeted than the known inhibitors, or the which are the known inhibitors of this pathway, or these enzymes of the glucocorticoid biosynthesis pathway. So, and this study was actually published in the Journal of Applied Toxicology, and it was the sole study, means there was no other study which was included in this paper. Only docking was there, which is telling the potential of the molecular docking tool, that if we are doing the pathway analysis, then we can find much more things that, which can tell, if not promising that they can be the absolute tools for that, but we can at least exclude the compounds, or we can filter the compound that we should proceed for the further studies or not. Second study, we have done the expression study of these same same enzymes in the adrenal gland. And what we actually did in that, what we actually found that these compounds we have taken, and we have checked the mRNA expression of both of all of these enzymes which you are seeing listed here. And we found that STAR, which is a very first enzyme of the glucocorticoid biosynthesis pathway, and this enzyme is showing that DVP and DHP is actually upregulating the expression of STAR, and downregulating the expression of CYP11B2, which is also an important enzyme in this pathway, and 3-beta HSD, which is also a very key enzyme. It is showing that these compounds are actually showing the increasing or the upregulation in this in the mRNA of this enzyme. And similarly, CYP21, 2A1 and CYP1B1, these are the two also enzymes which are also having the overexpression on the exposure of these phthalate esters. So this study was also published in the Journal of Environmental Pharmacology and Toxicology. So what we after all, what we did after that, we found that if these compounds, these are the enzymes which we have taken in our study, that is STAR, CYP11B2, and CYP21A1, and these compounds are actually connected with a number of proteins. So in this slide, I want to conclude that if the function or the expression, whatever may be expressed or may be compromised by any toxic chemicals, then the function and the expression, not expression actually, but at least function of these many proteins may be compromised in such a way. And I conclude that how many biological processes may be compromised if these enzymes may be the targeted, or these proteins may be the targeted. So I found that as many near about 300 processes in the body may be compromised or having the altered function, or maybe having the alteration in their function, if these enzymes of these, if the compounds are targeting these particular 5 or 6 enzymes only. So with minimum data, we can say that these enzymes, these compounds with the network toxicology, which I was showing in the last slide, with minimum data, we can say that a number of biological processes and a number of biological function and a number of biological organs may be targeted by these toxicants. So this was the this study which we are undergoing, and which we are telling that network toxicology may tell us a lot of things, along with which we are finding. For example, as the title of this conference is shown, that the alternatives to the animal experiment. So we may when we are designing our study, that we are targeting a particular enzyme or the particular pathway, then we should check associated with that how many other pathway, how many other how many other organs may be associated with that, which may tell us that we should proceed our study or not. So these many processes were listed here, that which may be compromised in if these 6 or 5 enzymes of the pathway or a single pathway is targeted. That was all. Thank you very much.
So the house is open for the questions. Yeah, ma'am, please.
Not humanist adrenal. Actually, I was studying the this particular pathway which is found in adrenal gland. For that, either I should have the cells of the adrenal gland, or I should have the primary culture of the adrenal gland, which was not that much possible in my in the available vicinity. So we have done that on the animal studies, and the purpose of this particular presentation was to compare that in vivo study with the potential of the network pharmacology, network toxicology. Yeah, in vitro assay we can do, but in vitro assay we should have particular cell lines of adrenal gland which is not available, and we have to either I should have the primary culture of the adrenal gland, or the these kind of cells are not that much available. Yeah, we have. Yes. No, this actually, as I said, this study was a part of my doctoral studies. So it was another project was which was associated. Nowadays, I am comparing that studies with the network toxicology. So it was having another project, so which we were looking for. So there are another aspect which I have to discuss if I need to discuss for these points. Yes, many studies are published and many studies are available with the adrenal toxicology of phthalate esters. But the prime worker William Harshay, which is working in the UK, he is also working on that. But the much not available on that particular pathway, which is I was looking for glucocorticoid biosynthesis pathway, and the whole that some studies were there on the steroidogenesis and some particular proteins and some particular mRNAs, but whole pathway of the glucocorticoid biosynthesis pathway was not targeted by Iman, which was our prime motto, because if one process is targeted, then that particular organ may be associated. And in the network toxicology, we are also having that if this particular pathway is available, targeted, then another pathway may be associated with that. So it was the purpose of. Yeah. No, it's in the first part of my answer, I answer your question that there was a data which is available still. My study, this study has been published 3 years back, sorry, 2 years back, and still the data on the whole pathway is not available. Yeah, yes, for that pathway analysis was the main target of that, and along with that, we were having some another studies, some another study which was that how it is related with the immune function. So some immune function which is related with the glucocorticoid biosynthesis secretion or glucocorticoid secretion. So those immune functions were also targeted in our study.
Dr. Shahzad, as I understand from your presentation, you carried out your animal experiments long time back.
Yeah.
Then now you actually wanted to see how much that toxicity which you got from your animal experiments is reflected in through network pharmacology.
No, not only that.
Network toxicology.
Yes, yes. Not only that, but also if I know already some data I am having, what else I can know with that? Because now, before I knew with the animal study that only glucocorticoid biosynthesis pathway was targeted, but now I can say that a number of pathways may be stimulated or targeted or associated with that, which may have the altered function with these exposures.
And to what extent do you think this toxicity was replicated in networking toxicology studies?
Sorry.
To what extent it is overlapping the toxicity which you got in animal studies? To what extent you found that toxicity in
So that
networking toxicology?
in the previous slide, if you can see this slide. This slide is showing that the even the what kind of potential is having. For example, if you will see this particular one pathway, in this pathway we will be showing that what is the strength. What if, for example, this including 10 proteins. So if 7 proteins are included in the network toxicology, then I can say 70% of the chances are there that it may have greater impact. If this pathway is maybe targeted, it is having the 10 proteins, for example, and if only 5 proteins are included in network toxicology, then I can see that 50% of the chances which they may have been targeted, or they may be targeted by these exposures.
That means about 50%.
Yeah, we can calculate that. Yes, we can calculate.
What about the toxicity of phthalates in children? I mean, that is also a very important point where, you know, the toys and other things which, you know, children, they put in their mouth, they all contain phthalates. And so have you gone into this aspect also?
Yes, sir. I have not the experimentally, but the epidemiological studies I have studied a lot. And I would like to mention you, when I was going for a international conference of IU talks, I was going to the US. So the embassy person has given me same question as you are giving me, that what is the impact of this on the children as well as different aspect of the population. So its importance, you can know that TSCA bill, which is available in the Senate of the US and the White House, so they were having a separate panel for the phthalate esters. So separate panel they are having means it is having a concern of compound. Along with that, number of countries which are banning the phthalate esters, especially in children toys, along with the well-known compound BPA, which Professor Isuddin, my professor supervisor, is working since long. So that these kind of compounds are already banned in children toys.
So much.
Thank you very much.
Thank you all. It was truly a wonderful session. The judges will finalize the winners and prizes will be distributed in the valedictory function. Now I request Professor Azhar Khan sir to present the token of appreciation to Professor Abul Kalam.
Thank you, sir. Now I request Professor Abul Kalam Nazmi, sir, to present the token of appreciation to Professor Azhar Khan, sir. Thank you, sir. Finally, I hand over the podium to Dr. Ajit Xavier and Dr. Hiramani Rava. We already told.
Good evening, everyone. I, Dr. Hiramani Rabha,
And I, Dr. Ajit Xavier,
Junior Residence, Department of Pharmacology, we are honored to introduce Dr. M. Isbauddin sir and Dr. Christian Pellevoisin sir, who will be present online as the chairperson for this session. Dr. Christian Pellevoisin sir, who is PhD, is a distinguished scientific director at MedTech Life Sciences, France. with rich background in toxicology, biotechnology, cosmetics, and medical device industries. Skilled in research and development, toxicology, in vitro assay, Sir has been instrumental in developing innovative reconstructed human tissue models and in vitro models for assessing product safety across diverse industries, including cosmetics, medical devices, and pharmaceuticals. His unwavering commitment to advancing alternative methods of animal experimentation and his role in developing, validating, and standardizing in vitro techniques make him a pivotal figure in the field. Additionally, his dedication to knowledge and sharing education, particularly in the realm of predictive toxicology, further cements his status as a leading expert in the industry.
And now I would like to invite Dr. M. Misbahudin, sir. Sir is an alumnus of JNMC and presently works as an associate professor of pharmacology at the Faculty of Medicine, King Abdulaziz University, Saudi Arabia. He has more than 65 research publications and serves on editorial boards of several journals like International Archives of Biomedical and Clinical Research and Advances in General Practice of Medicine. He also serves as an academic editor for PLOS ONE and associate editor for the Journal of Biomedical and Life Sciences. Dr. Misbahudin is affiliated with the Faculty of Pharmaceutical Medicine by the Royal College of Physicians, London, and the Royal College of Physicians of Edinburgh. Sir has shared his expertise in myriad scientific events across the globe and has received several awards, for example, Best Paper Award at the World Congress of Diabetes, USA, and also Citation Prize from King Abdulaziz University and Excellent Leadership Award from IDB at Saudi Arabia. His research mainly focuses on identifying novel genetics variants, lipid pharmacogenomics, biomarkers, and other exploration of novel therapeutic targets through in silico protein ligand docking and molecular dynamics studies. Now I request Dr. M. Misbahudin, sir, to introduce our distinguished guest and speaker, Professor Nobuhiko Kojima, who will be delivering his lecture online. And Professor Nobuhiko Kojima is also representing Japanese Society for Alternative to Animal Experiments. Over to you, sir.
Thank you very much, Dr. Ajit, for your nice words. Let me introduce two learned resource person for today. Professor Nobuhiko Kojima. He is an academic researcher and associate professor at the Department of Life and Environmental Sciences at the Graduate School of Nanobioscience of Yokohama City University. Professor Nobuhiko contributed to research on cell culture, tissue engineering, disease models, stem cell differentiation, cellular spheroids, and 3D cell culture. He is currently holding a CTO position at EcoCell Company Limited. Professor Nick Jukes, the second resource person, he is the coordinator of InterNiche, the international network for humane education, and he has been working internationally for over 30 years to enhance education and training in medicine. Not only medicine, he also works in veterinary medicine and the life sciences through the replacement of animal experimentation and dissection. Professor Nick co-authored the book From Guinea Pig to Computer Mouse and has published widely on alternatives. He is the coordinator of InterNiche, which is an international network for humane activity, which has led to widespread direct replacement of animals in experiments. Prof. Nick, the InterNiche and its partners have won several accolades for providing resources, global networking, and the replacement work, including the Lush Prize for Training. So I welcome Prof. Nobuhiko and Prof. Nick Jukes today on this platform. Welcome. Thank you very much.
My name is Nobuko Kojima from Yokohama State University. First, let me introduce Yokohama State University and myself. Where is Yokohama State University located? Haneda Airport is here, and this is Shinjuku. So Yokohama is located in south of Tokyo, and this picture shows an urban image of Yokohama. However, main campus of Yokohama State University is a little bit far from Yokohama. We need 20 minutes by train from Yokohama to Yokohama State University. It is very local atmosphere like this, completely different from the center of Yokohama. But the campus is close to station, just 5-minute walk, and close to shopping center, barbecue site on the beach, aquarium, and small yacht harbor. So I think our campus is very local but useful and beautiful. This is a picture of barbecue party with Labo member. There is a beach here. There is a beach. So we had a barbecue party just 2 months ago. Yokohama State University is a small university. We have just 5 undergraduate schools and 6 graduate schools. I belong to the School of Science and the Graduate School of Nanobioscience. The number of total students is just 5,000, very small. There are 758 teaching staff, so we are small, but the distance between students and teaching staff is relatively close. The main theme of our laboratory is 3D culture of animal cells. We can design various structures inside of the multicellular spheroids. These special spheroids are powerful tools to realize alternatives to animal testing. Okay, this is today's agenda. I'd like to talk about 3 topics. The first topic is introducing of 3R's promotion committee. The Japanese Society for Alternatives to Animal Experiments, JSAAE, had 8 standing committees: General Affairs Committees, Planning Committee, Editorial Committee, Public Relations Committee, International Exchange Committee, Finance Committee, Academic Committee, and 3R's Promotion Committee. 3R's Promotion Committee has 3 main activities. One of them is delivery lecture. We visit university and give lectures on 3R's. The target of the delivery lecture is mainly university students. Roundtable talk is another activity. We talk with company workers about 3R's. The target of this activity is company workers. And the last one is the challenge contest. This is for high school students, especially senior high school students. This is a roster of 3R's promotion committee. I am the chair of the 3R's promotion committee. Vice chair is Tadashi Uchida. Advisor from the board of the JSAAE is Masaharu Akita. And there are 12 members. In particular, the members whose names are in red contributed greatly to the challenge contest. Let me introduce Professor Masaharu Akita and Professor Yuji Nashimoto, Professor Ikki Horiguchi. They are from university. Mr. Taro Kobayashi, Ms. Ayano Tokunaga, Miss Miki Asanagi, and Miss Minori Mezaki. They are from company. They worked very hard for the challenge contest. I'd like to take this opportunity to express my gratitude. Thanks so much. Let's move to the next topic, concept of challenge contest. What is a challenge contest? This is a contest where high school students learn about the 3 R's and compete for suggesting an original method about alternatives to animal experiments. Why do we target high school students? Active learning of the 3 R's for high school students is extremely important to develop the human resources of the next generation. Now, I will explain the history of challenge contest. Challenge contest was started in 2006 and held for 8 years until 2013 at the face-to-face venue. The number of students participants were 18 and 15 at the 7th and the 8th contest. For 8 years, the event was successfully held. However, the contest was interrupted by a problem that a general participant made a negative comment against high school students. The 3R's promotion committee took this situation seriously and decided to cancel the challenge contest for the time being. Last year, 2022, the JSAAE changed from a voluntary association to a general incorporated association. This change triggered the decision of the President Hiroshi Itagaki. He ordered us to resume the challenge contest. We just said, yes, sir. From 2006 to 2013 is a kind of first period of the challenge contest. In the 88 years from 2014 to 2021 is interrupted period. So from 2022, it can say that second period of the challenge contest. This is a general outline of resumed challenge contest. Idea-only proposal for an alternative method is acceptable. The prizes will be determined in 2 stages, document review and online presentation. Participants are limited to related persons, students, board members, and sponsors only. It means we excluded unknown participants to protect high school students. To resume the challenge contest, advices from Mr. Kodama were very helpful. He is a high school teacher and a former member of 3R's promotion committee. He advised us that high school students and their teachers are extremely busy. So, date of the contest, a style of application form, presentation time were well discussed. And this discussion reduces the barrier to entry. Another advice was that personal information of students should be highly protected. So we carefully treated their personal information, name, photo, and it reduces unexpected troubles. The final topic is how to run a challenge contest. We asked a professional event company, CTU, Corporation Limited for support the challenge contest. They supported management of applicant information corresponding with applicants, operation of online system on the final presentation, and making a leaflet reporting the content. This slide shows the schedule of Challenge Contest 2023. The end of last year, November 2022, we started looking for sponsors. These companies are the sponsor of the Challenge Contest 2023, and they each provided 100,000 yen, so we obtained 900,000 yen from sponsor company. I think the total cost is about 1.4 million yen. Therefore, the shortfall was covered using budget of the JSAAE. Of course, we need to keep effort to find more sponsor companies to reduce the burden on JSAAE. April 2023, we sent letters to high schools to let them know the challenge contest. Application period was started on June 1st and ended on June 13th. And first screening by document review was held in early July. In Challenge Contest 2023, there was 17 entries from 10 high schools. Total number of participants was 31. The number is higher than last year, 2022, just after resuming, and also higher than 2012 and 2013 before interrupted. The result of the first screening was sent to participants in mid-July. We selected 5 groups. The final presentation was held on August 18th and decided the awards. CTU Corporation Limited set up the operation headquarters on the day of the final presentation. This room is the meeting room of Ikeda Rika Corporation Limited. Because Mr. Kobayashi is here. Mr. Kobayashi, a member of 3 Arts Promotion Committee, belongs to the Ikeda Rika, and he negotiated with his boss to use this room and their facilities with no charge. Fortunately, we were able to use this room and their facilities in 2022 and this year, 2023. The president of Ikeda Rika Corporation Limited, his name is Takahashi, visited us on August 18th and said this event challenge contest is very important, so you can use our facilities next year too. I said, wow, it's very lucky. So we are able to hold the challenge contest smoothly with the support of various people, including the president of Ikeda. Let me introduce the presentation of high school students. Ms. Izumi Fujimoto and her colleague got the first prize. The thesis is sea squirrel as a substitute animal. Second prize went to Ms. Yuzuka Sasaki and her colleague. Ms. Sasaki is the only junior high school student among the participants. Their thesis is alternative to animal experiments on cranial nerves using sea squirrel. And another second prize was given to Mr. Zen Kaneda from Hibarigaoka Gakuen High School. His thesis is possibility of animal experiments using Spiona intestinalis. Spiona intestinalis is a kind of family of the sea squat. Why were there many species using sea squat? This is because sea squat is evolutionarily close to human. Miss Nonoka Takada and her team member got third prize. Their thesis is stimulus evaluation by plasma flow velocity of Brazilian water wheel. Another third prize was awarded to Ms. Akane Takada and her colleagues. The thesis is endotoxin detection method using artificially cultured horseshoe plant blood. After first screening in early July, we sent some comments to improve their study and presentation. The students seriously revised their presentation. We felt that interaction between high school and university or company is very synergistic for their education. We prepared the report leaflet in October. This is the leaflet. It has 4 pages. There is information of title here and sponsors here and thesis from students here. And this part introducing the overall review. And this part is about awards from sponsor companies. The sponsor companies chose a presentation what they were especially interested. And this part shows awards who was not able to pass the first screening, but it has good ideas. In the next week, from November 27th to 29th, the annual meeting of the JSAAE will be held at Chiba University. The ceremony for the Challenge Contest Awards will be held in the annual meeting. And the students will show their presentation in a poster session. The Challenge Contest was an online event. However, some of the winners were invited to the JSAAE annual meeting and awarded again. This picture was taken last November. And this year, almost 10 students will attend the annual meeting in the next week. The students obtained the prize will present their study at a poster session. Their presentation was very nice for the JSAAE members. It was really moving us. Everyone involved in the challenge contest felt that it was very hard event, but it was really valuable. The effect of the challenge contest. The contest is not just for education of high school students. All members of the JSAAE who were related with the challenge contest were stimulated by the presentation and behavior of high school students. Several persons of sponsor companies joined the JSAAE immediately after the challenge contest because they were highly impressed by students' presentation. This event is very effective for adult members and it can affect all stakeholders of alternatives to animal experiments. This is summary of this presentation. Challenge contest is an educational event for high school students. The JSAAE held the contest from 2006 to 2013 and resumed it last year. The contest was felt to be meaningful and worthy for all stakeholders. So next year, 2024, we would like to hold a challenge contest again. Thank you for your attention.
Thank you very much, Prof. Nobuhiko. It is really very nice to see your real efforts, and you are involving students from the very beginning. I mean, high school students, if they are aware from the very start about this important issue regarding the alternatives to animal, is really something which is going to bring a change. No? Because mostly people, when they are in graduation or post-graduation, they go for research. But you are inculcating this idea of alternatives from the very beginning. So I'm really very impressed by this real work, actually. Thank you very much once again. Now I open the session for questions.
I have one question, if I may. Christian Pelvoisin speaking from far. You hear me? Do you hear me?
Yes, we can hear you.
Ah, thanks. Yeah, great presentation. I'm very excited by this approach to try to engage young students early in their career development, to give them an awareness about alternative methods. So that's very great. I have a question for Dr. Kojima. From the previous contest, have you got some example of methods that reach a real development and that are used now in some laboratory? Or if it is not the case, do you think that in the future, with the new this new contest, this could happen, that some student propose a method and that this method will arrive maybe up to JAGVAM validation in the future? Dr. Kojima, do you hear me?
Prof. Nobuhiko, can you hear Prof. Christian?
No.
Sorry, Prof. Christian. Prof. Nobuhiko is not here.
Anyway, I like this presentation, and I think we can learn from what has been done in the future to stimulate also in Europe and maybe in India, to motivate some young students to start to work on this area. Thanks. Thanks, Dr. Kojima.
Thank you very much once again, Prof. Nobuhiko. Now I invite Prof. Nick Gibbs, the second resource person for today, to start the session. Thank you very much.
Good afternoon, everyone, and I'm very happy to be here speaking at your conference today. It's been a while since I've been in India, but I used to be a regular visitor, so at least I can make it virtually. What I'm going to do today is to talk about humane innovations and replacement of the harmful use of animals, such as animal experimentation, in higher education. So in the classes and the practical classes of veterinary education and training, medical education and training, and biology. I'll be giving a review of the background of the situation, the many different types of alternatives and humane innovations. We'll be watching a summary clip of a surgical training tool with its many clinical skills and surgical training functionalities, which is part of a film series that we're producing. And finally, I'll be looking at some of the wide advantages of using these tools in terms of the quality of education and training, as well as the ethical and other issues for the benefits too. And then I'll be concluding. So it'll be about a third talking, a third film, and a third additional talking before the questions and answers period. So I'm now going to share my screen and go to my PowerPoint, and we'll be talking through that. So let me just enlarge this. And go to share screen. Here we are. And here's my PowerPoint. So hopefully you can all see that clearly. Okay, so an introduction to alternatives to animal experiments as the broadest term in education. Just a little taste of what some of these humane innovations are. This is a virtual laboratory following one of the classical frog muscle experiments for teaching physiology. And this is something relevant for medical, veterinary, and biology education. And this is a high quality virtual laboratory, which is highly interactive and very useful for teachers and students. Just a shot of one of the laboratories. This is the Sindava canine. This is an advanced clinical skills and surgical training mannequin that has been developed in the US and is now used increasingly worldwide. And we'll be looking at the film of some of the surgeries on this tool in the film clip later. But as you can see, there is a enterotomy, resection, and anastomosis laboratory being done at the moment on this synthetic dog. This is a Dutch body donation program for ethically sourced animal cadavers, a national program that, again, like the other tools I've just shown you, I'll be explaining later in more detail. But this is a body donation program for ethically sourced cadavers as replacements for dissection within veterinary medicine. And this, of course, is a typical operating room shot where you can see professional surgeons being guiding and working with younger students and trainees in an apprenticeship model, and of course working with a patient as long as the student or the trainee has sufficient skills and is prepared for it, is also an alternative to an animal experiment within surgery. So let's begin with some of the context and the definitions regarding this field. Firstly, we can look at harmful animal use or the harmful use of animals in a number of different fields: education and training, which is my focus, research, and testing. Education and training, however, is a unique field. It differs from pure and applied science in a number of ways. Firstly, it's not research, which is the acquisition of new knowledge rather than known knowledge. Nor is it testing of drugs and chemicals for regulatory purposes or for applied research. So it's a pedagogical angle. And while there are connections between all the fields, it's important, I think, to be able to differentiate between them because of their many different natures and how we can apply new innovations to those fields and what is the purpose of those innovations. So I think we can agree that humane education and training is a prerequisite for humane research and testing, familiarity with alternatives, and also better quality education and training too. Also, there is continuing education within the professionals, within the professions as well. So I think we can recognize that education and training is not only for students, but also continues throughout the duration of a career as well. Now, there's three areas where we can talk about education and training. The first is education at secondary level, higher education level, and the professional level through the use of alternatives. So alternatives are the tools for that education. And this is what I'm going to be focusing on in my talk today. Then there is training in alternatives for application in research and testing. And this is a rapidly growing field where a lot of people are training scientists, technicians, and assessors in different NAMs, or non-animal methods, new approach methodologies. There's also education about alternatives for students and for society, as we've just heard from the previous speaker. So at the secondary, higher, and professional levels, we have education of students and trainees through the use of alternatives in order to gain knowledge and skills and attitudes. So alternatives are the tools for the process of education itself. So it's a pedagogical issue, and it also involves addressing, acquiring, and mastering technical skills. And replacement can be achieved within this area, of course, and increasingly is and has been. So when we look at the educational structure, as most of you will be aware, of course, at these levels, in higher education and professional level, we have different faculties. Sometimes there might be harmful animal use within psychology, dentistry, and other areas as well, but these are the main areas. Within each of these faculties, we have the different disciplines, and within each of these disciplines, we have the practical classes and the training events where we may find harmful animal use or may find humane, innovative tools. Now, the majority of conventional animal use has indeed been harmful. It involves, or has involved, dissection of animals killed for the purpose, animal experimentation, and other instrumental animal use, which doesn't fit into either of the first two categories. But I think it's also important to remember that there are also humane traditions. This might be by individual country, by discipline, or by practical class. A teacher and a faculty may have always done their physiology practical classes on student self-experiments, for example, looking at exercise physiology or respiratory physiology. That also might be the case in some countries. Similarly, veterinary surgery in the UK, where I'm from, is generally learnt using non-animal methods and then work with animal patients, helping them as the students get apprenticed into the different areas of surgery. So we are looking at replacement and full replacement, where harmful animal use might continue in some disciplines or some departments and some faculties. But we should also recognize that sometimes there are traditions that have always been humane, which of course can benefit further from new innovations, but which don't have the same ethical challenges as animal experimentation. And there's also widespread ongoing replacement and progress towards humane education. I had a meeting recently with a university in Hong Kong, a virtual meeting, and I understood that virtually all the work with animals for education was humane, either a body donation program or work in the clinic, or use non-animal methods. So sometimes we find whole departments, even whole faculties, where the humane innovations or the positive traditions are providing a fully humane, a fully ethical education already. So it's very exciting, I think, at the moment within education to see such progressive change and to see the focus on teaching objectives and how they can be better met using these humane innovations. If we're talking about replacement of harmful animal use, we need to briefly define harm, of course. So we define it as any action that limits the following freedoms. Freedom to live, because killing is a significant form of harm. freedom from pain, injury, and disease, and other freedoms that we define in more detail within our policy at our website, interniche.org. Now, this is quite a strict definition of harm, but harm is a serious issue. So I think it's very important to look at it critically and to find solutions so that such harm can be reduced to zero, not just minimized, but reduced to zero. And that's what we're going to explore through my talk, including with case studies of where it's already been achieved at some of the most prominent universities around the world and at many others too. Harm, of course, is not just a question of what happens to the animal. Is he or she killed? Is he or she experimented on? When you look at the whole picture, the whole life cycle, we can see there are broader animal welfare and ecological aspects associated with harmful animal use that we need to challenge. There's capture or breeding, transportation, caging and keeping, the experimentation and killing itself, preservation and disposal. And within all of these, we have issues that we can address. Now, many universities are introducing courses about ethics, bioethics, animal ethics, about alternatives and the 3 Rs, and about animal welfare. And this is very important. I think it's important to add to education, to enhance it. However, what happens when education about these issues is also going on in parallel with harmful animal use? And we find, I think, that teaching about those issues whilst also practicing animal experimentation is inconsistent. And the lessons of that education might be negated by harmful animal use. So I think it's important to look at the use of replacement alternatives in education and training, and to recognize that and to the achievement of that is also a significant part of the alternatives education put into practice, the animal welfare education put into practice. So these two things really need to be parallel. We can also look at the 3 Rs more specifically and ask, are they helpful or even relevant for education and training? Now, the 3 Rs derives from a laboratory animal science, an LAS perspective, rather than a pedagogical one. So I think there's limited relevance here. Even so, within the 3 Rs, replacement is indeed prioritized and is the actual objective. So I think we can redefine the 3 Rs for education and training and recognize that from the examples we can see around the world of replacement, full replacement that has indeed happened, that it's feasible in education and training, and that 1 R, not 3 Rs is better. So making them stricter. Similarly, making them smarter, finding replacement solutions that might still involve animals. It doesn't have to be a non-animal approach. However, working with animals, it needs to be fully humane and have zero additional harm. And that doesn't mean refining an animal experiment or reducing it. It means no animal experiments, but finding ways, for example, for a body donation program to provide cadavers for veterinary anatomy, and for veterinary teaching hospitals to have greater capacity, well, to exist, firstly, some places don't have them, so to exist and have greater capacity for students to work with animal patients and to learn and further enhance their surgical skills and their clinical skills with animal patients who benefit from that interaction. So in both of those cases, they are both fully humane. And finally, we can think perhaps of retiring the 3 Rs and for education, focusing on enhanced education and training and looking at teaching objectives in the broadest sense, which is what we're going to be looking at further within my talk. NAMS, for example, new approach methodologies in research and testing, is a similar way of where enhancement of the science is also bringing about that replacement. Now, sometimes we find the phrase "the real thing" appears. It's a myth that I want to address. The myth is that animal experimentation is somehow the real thing, and that alternatives or humane innovations are not. But I think this confuses the method with the objective. Animal experimentation is just one method, and the real thing is not the animal experiment. It is effective gaining of knowledge and skills and attitudes and positive approaches as well. So that's the real thing. So the key points from my introduction are that we're addressing curricular design, we're defining teaching objectives, and the choice of tools and approaches that can enhance the acquisition of knowledge and skills and attitudes. Let's now look at the different types of alternatives that exist, or that could be further developed indeed. Alternatives are tools and approaches that can replace harmful animal use and meet teaching objectives. And in many departments, these alternatives are now the norm. And in fact, the word alternatives is often no longer even used, because these are now the standard and better ways of teaching. We can look at non-animal tools, and we can look at the fully humane approaches that involve animals, as I've explained, like working with animal patients. And we can look at the different categories of these alternative methods: film and video, computer simulation, augmented reality and virtual reality, models and mannequins and simulators, ethically sourced cadavers, organs and tissue, clinics with human and animal patients and animal volunteers, self-experimentation, and we're going to look at each of these in turn. Firstly, film and video, of course, such as professionally performed dissections, which can provide a very, very good tool for students to learn anatomy without any mistakes, of course. And I think we can recognize both the benefits of that and being able to work with digital video, as well as the limitations. So, for example, video is not hands-on. It doesn't meet that group of teaching objectives. However, there are other ways, of course, of reaching those objectives of having hands-on skills. And like any teaching approach, there are multiple tools and approaches that will be brought together into a framework in order to meet the range of teaching objectives that has been defined. So the fact that one tool doesn't have a certain opportunity doesn't matter, because you'll be using a second or third or fourth tool in addition, and together helping to meet and in fact often exceed those existing teaching objectives. Computer simulation has a number of different areas. Firstly, there is the virtual dissection, often with different magnifications, labeling, background information, ability to compare between species, very, very flexible, very helpful for learning anatomy. There's also the digital anatomy program based on photographs. This is from the virtual canine anatomy program developed at Colorado State University in the US. And this is a stills image from one page within deep dissection of the head. And you can see there's labeling on the image, and there's labeling information in the menu, as well as additional information and hyperlinks at the bottom. So you can both click on a nerve in the menu and have it highlighted on the image, or you can click on the image and find out what that muscle or vessel was. Now, as you can see here, we have dozens of labels, all within one image. And that image is one of 12 images showing the different levels of the deep dissection. And the deep dissection is just one of the dissections. There's also superficial dissection. And the head is just one of the structures that we can look at the anatomy and also the radiology for. So you can see that this is an extremely complex and rich learning tool with thousands of labels and images in anatomy and radiology that you can use for the student to learn and to revise and to go back and forth and really seriously enhance the quality of the anatomy lesson. This is also available in a virtual reality model for the students at that university. This particular tool is available for free, and you can contact Intl to get advice on receiving a copy of this as well. Then there is the anatomy program where you can dissolve away different layers of the structure of the body, different structures, in order to be able to highlight and see at different levels. And this is a different type of computer program because it doesn't simulate dissection or have photographs of dissection. Instead, you can go deeper and come back to more shallow levels in the body. And I think that's a very good way of visualizing structure and structural relationship. Then there's the virtual laboratory, very good for physiology and pharmacology, where you have dynamic systems change over time and the need to be able to correlate between different events. So this is SimNerve, this is SimMuscle, developed at the Institute of Physiology at Marburg University in Germany by professional physiologists working with programmers and graphic designers. And this simulates these simulate the classical frog nerve and muscle experiments, but does it in a way where you have algorithms producing the results, which are diverse and account for biological variability, and occasionally even the apparatus, the virtual apparatus, breaks down as part of the range of lessons for students. Sometimes in a lab, equipment doesn't work, and it's good to also experience that in the simulation too. But the teachers, most of the time, of course, it does work, and that's what you want, but the teachers can also monitor the students' results, and the students can work at home and analyze further. So again, it gives a lot of opportunity for self-directed work and for students to work in teams and to be very good at gaining knowledge within physiology, understanding physiological principles. This, in combination with self-experimentation, is excellent. First, you gain to understand all the principles, and you really, by going back and forth, you have the chance to really, as a student, make sure that you have understood the principles and whether the teacher thinks you have as well. And then you can apply those to the living body. And of course, the human body is an excellent object for you to be learning on as well in a non-invasive self-experiment. So those two together are a very, very good partnership for learning physiology. You have animations in computer simulation as well. For example, this is from the glass horse equine colic software, which shows how different structures exist and where their positions are, but also how different conditions arise and how you can do examinations to identify those conditions. And you also have radiology, ultrasound, and so on for you to be able to build up the quality of your diagnosis work and understanding of conditions. So that's another type of software. And of course, you have many apps where you can revise and practice different clinical skills and surgical skills in a virtual way, or also anatomy too. At the more advanced and expensive end of simulation, of course, we have augmented reality and virtual reality. Augmented reality is semi-immersive. You're using real objects and a tablet or goggles to be able to understand different structures, but also clinical skills and surgical procedures too. And then virtual reality being more advanced, where you have a very immersive sensory environment, which may or may not include the sense of touch, haptic technology, which simulates the sense of touch. And this is on the left, you can see an anatomy program. On the right, the haptic cow, using a model and a computer simulation to provide a very rich virtual reality experience to contribute to your work in diagnosis of clinical skills and surgical procedures as well. Models and mannequins and simulators is the next area. And after this section, I'll be showing you the film clip, which can explore advanced surgery for dogs in more detail. So models have been used, of course, for hundreds of years. Plastic, wooden models. Every country has producers of models. They can be excellent for understanding anatomy. As you see here, a paper mache model of the human body from 150 years ago. These tools have been used for a long time, and they're still used because they have value. This is a horse model that you can take apart. This is using clay to build up the musculature of a horse on a model skeleton. And this creative approach to learning anatomy, this slower approach, this hands-on approach, I think, is very valuable as one of many tools that can be used for getting a comprehensive understanding of anatomy. You can also have 3D printing for local production of teaching tools, from bones to soft tissue, and that's a very exciting area that's developing very fast. And then you have models produced in factories, often with many layers, sometimes with resting or circulating synthetic blood, such as this suture pad and model that has 5 layers. And this is very important as a basic model for basic surgical skills. It's a low fidelity model, a low level of realism, but it's of sufficient fidelity for the specific skill set at the early stage of learning surgery. Because, of course, the practice of surgery for vets and for medical students is something where you are continuing to build up your level of skills and the complexity associated with those skills and the procedures required. And so basic models at first, intermediate, advanced models towards the end are all part of the process of developing surgical skills. At the end, of course, you'll be working, we hope, with human and animal patients in a way that helps them, but the non-animal methods can range from very simple to very advanced, and they're all relevant, they're all good. This is a rabbit ear model for practicing taking blood and giving injections in the rabbit ear. This is a spay-neuter model for dogs, where you can practice this in advance of working with animal patients. This is some photographs from a surgical skills lab in Denmark, where the teacher uses bought soft toys and then dissects them and adds very simple elements to those toys in order to produce models of sufficient fidelity for basic surgical skills. So this is using balloons, condoms, rubber bands, mayonnaise, other simple and inexpensive materials for teaching a range of different methods for which this level of fidelity is sufficient. So you don't need an extremely expensive model. I'm going to be showing you a clip of an extremely expensive model to show you what can be done, but do remember that extremely cheap and sometimes free material is indeed available. So local production of material is also possible too. So this is one of the more advanced models. This is the Sindava human, and we're going to be looking at the dog version of this in the clip. Here we have the bleeding liver and spleen. This is looking at a foreign body within the intestine. This is one of the skills labs that I was filming at. And here you can see the enterotomy being practiced by the students in a very controlled way. So it's a very good environment for practicing. No major ethical problems that confuse the learning process, no additional stress that will do the same, but a big, good focus on specific procedures after basic surgical skills have been learned with other models. And of course, processes and procedures using these models can be learned as well. Anesthesia, other models. This here you can see the other types of models that are used. Anesthesia here for the dog. And this is another dog which I brought in the past to India, in fact. It's called Jerry, dog model. And in this case, the veterinary professor has used a second-hand heart pump, and he creates scenarios with case studies of different canine patients for students to be able to diagnose the problem and apply the right drugs to help solve that problem and save the dog. So in this scenario, 2 students go into this room and work with this dog according to the different symptoms that are presented, that the teacher is controlling, and the students can read about and watch on the dog. And in another room, there's a group of more students who watch what students are doing. And then afterwards, all the students come back together and discuss what those 2 students did in a very non-judgmental and inclusive way. And then it swaps around to other students with a different case. So this is really excellent simulation for a wide range of different processes, procedures, and of course conditions. So there's a lot that can be done by building up your own tools, using bought ones and enhancing them further, like in this case. And there's a great number of new skills labs for clinical skills and surgical skills that are being developed around the world, such as this lab at the College of Veterinary Medicine at the University of Florida, where also I was filming. So now let's have a look at this clip, so you get a chance to see what some of the most progressive universities are doing with one of the most advanced surgical training models. And you can have a break from me talking by listening to some other people, watching the demos and watching the labs. And this is a 20-minute clip, after which I'll be concluding my talk with an exploration of the advantages and some of the alternatives and some of the resources that we can offer.
My name is Dr. Galina Hayes. I'm an assistant professor at Cornell University. I work in the soft tissue surgery section, and my responsibilities include providing service to clients and animals coming to the hospital either on an elective or an emergency basis requiring soft tissue surgical procedures, as well as training the students, both the 3rd and final year students, in the theory and practice of surgery. Veterinarians are expected to know and do a lot and have a grasp of a very wide range of skills, which in my opinion may not be entirely realistic to expect them to accomplish within 4 years. Their exposure to hands-on surgical training may be limited to just a few weeks in their final year of their DVM training. And within those final weeks, there may not be that many opportunities for direct mentorship. In the veterinary world, we leave and are expected to be surgically competent at the end of just our basic DVM training. And while I don't expect that to change anytime soon, I think we can change the way we deliver that training through the DVM program, to try and make sure that the skill set that our graduates are equipped with is a little bit closer to what they're expected to have.
Within the sophomore surgery course, the course that I coordinate, I have tried to focus on clinically important, practical concepts, techniques, knowledge. What are going to make a difference for my patient? For example, GI surgery, very, very commonly performed. Dogs eat rocks and toys and they plug up their guts and then they need to have surgery. If you don't do the surgery correctly, there is a good chance they're not going to survive because they're going to leak the contents of their bowel into their abdomen. I'll spend a lot of time talking to them about how do you properly assess the bowel to make sure that you've got healthy tissue that you're bringing together, and how do you suture it together correctly, because you can suture it together incorrectly. You spend a lot of time doing it, feel good, and have a failure. I do believe that if you understand the tissue properties and the pathophysiology of the disease, and you have basic surgical skills and common sense, you can apply those basic skills that you learn from your courses and maybe a clinical SPAE to other procedures.
In my opinion, surgery is a very it's heavy on hand skills, yeah, and you can talk about it in a PowerPoint all you want, but nothing really replaces having someone stood across from you, adjusting how you're holding instruments, adjusting your body position, adjusting your tissue handling, and it's those skills that translate into patient benefit and patient safety.
So here at Syndaver, what we're looking for is to fulfill curriculum need. University of Florida specifically saw the need for an alternative to cadaver surgery. It was not working. And live surgery, too, was something that they did not want to persist with. They had specific curriculum tasks, surgeries that they wanted the students to be able to do that we needed to fulfill in the model.
We set up an interview meeting with them. We drove down to Tampa one day. We pitched an idea to them about using their technologies that they had already, developed and had been marketing and applying on the human side for dogs. From a practical perspective, the vascular anatomy. So how big is the vessel? Where is the vessel? Is it there or not? Does it connect to the right places? With the texture of the organs, are they stiff, too stiff? Are they too friable or too soft? And now we spend a lot of time on them and they're beautiful. The liver is very, very close to the texture, the feel, the compressive abilities, qualities. It's vascularized. We have a little bit vascularized of a dog liver. We've got quite an impressive high fidelity canine.
So the first thing that I'm proud of and like is the fact that it does have a very realistic airway. The tongue, as well as the internal aspects of the glottis and all, allows intubation. Also, the face, I try to give it kind of kind eyes, and you feel a bit of empathy for the canine as you use it. Second, we do have the ability to put IV in both front legs in the cephalic vein. To that, we can also draw blood from those areas. We have jugular veins. It is modifiable, so it allows central line catheterization, but you can draw blood from both sides. It also is a pulse, and you can create anesthetic scenarios where the learning experience can be beyond just what's being done within the dog. In the real world, you've got to worry about their breathing, you've got to worry about their blood pressure, their heart rate, and the dog, because of our ability to have it kind of change through a tablet, we can alter heart rate. We can put them in ventricular tachycardia. We can modulate them. Looking into the chest cavity, there is a heart, as well as you can see both lungs and the bifurcation trachea. Since this can be intubated and hooked to a respirator, the lungs will expand, and you can, of course, adjust that to enhance the scenario.
Conceptually, what we do is very simple. We start out with a live tissue study. So we'll actually get a sample of a tissue. We'll run it through a battery of tests that can be mechanical, physical. They can be things like puncture resistance, coefficient of friction, abrasion resistance, tensile modulus. Stretching, pushing, pulling, and measuring the kind of loads you get in response. Then using a knowledge of material science, you can come up with candidate materials to try to mimic all of those things. And you might have 12 different properties that you're trying to mimic. And through a process of experimentation and designed experiments, you can come up with a material that would be considered a validated mimic for skeletal muscle, for example.
And, once you've done that 100 times, you've got, materials that you can build an entire body with.
Certainly, I think one of the greatest strengths of Sindava use is in its ability to provide a very high fidelity environment for the practice of abdominal surgery in dogs. For me, the intestine in the Sindava is the closest to live animal dog intestine that I've been able to find. It has very similar layering, it holds suture in a very similar manner, it handles the same, you can perform the same procedures in it, you're working down within a cavity, it holds staples in the same way that live dog intestine would. We've really tried to focus on two target audiences, if you like, there's general practitioners and ER doctors. Probably the highest risk procedures that surgeons in that setting perform are intestinal procedures, and there's a intentional emphasis on intestinal procedures for those reasons. So the students are assigned in pairs to a cadaver unit, and they each alternate performing different components of the procedure. And then our focus during the lab is to teach them gentle tissue handling, efficiency in their use of instruments, how to maintain a flow of productivity through the procedure and not get bogged down in unnecessary snarls, and safety, how to maintain a sterile field, how to manipulate tissue so they can maintain good visualization. And it's those core skills which they can then take on and apply to pretty much any other procedure that they try and do in the future, but which are impossible to teach in a didactic setting.
This leads us to the bladder. And of course, the bladder specifically has a few pathologies itself. There are, in this case, 3 stones that are palpable within it. This, again, guides the surgeon to perceive there to be a problem. There's stones there. He doesn't know how many. So he must open the bladder, remove the stones, but thoroughly pass a catheter, clearing it, and then passing it again.
You can either flip your blade upside down, and just like the linea, do the reverse stab incision up, and then extend with a pair of scissors, or you can paint down with the blade until you're in the lumen, and then extend with scissors. But I think either of those are fair. Ready. So, when you guys are suturing the bladder, please ensure again you go serosa to mucosa, mucosa to serosa to ensure that you engage the submucosa. Use your needle here to grab the loop. Pull up, right? That's going to tension the previous sutures that you placed, and then pull it down. And as you pull it down, you're going to be able to put it in the position that you want the suture to lay. And then once you're there, try not to move the knot, and then it's another additional 5 throws here, so 3 total square knots. And I like the way you're flattening your hands. Rock your hand and your needle driver in your hand as you go. And so as you rock it, it's going to tighten each side now.
Going further in, we would exteriorize the intestine. And again, as a good surgeon, you are thorough in your exploration and will feel of the intestine and determine that there are certain irregularities here that you don't want to miss. So it feels kind of like a dice. How would I deal with this particular thing? Well, you would do an enterotomy. So at this point, you would cut, remove that foreign material, sew it up. But there's another issue here too. There's a pathology, an area of irregularity, perhaps damage that was done by the dice. So this would guide us to consider this area an area where I would do an anastomosis.
What is going to be the preservation of your blood supply? And this is the hardest portion to figure out, I think, with a resection anastomosis. Twofold is where to cut and what blood supply to save. And so, again, if we're pretending that we need to remove all the sutures of the enterotomy, what arcades need to go and what arcades need to stay? You have great blood supply from this jejunal arcade to supply this from this end. You want to actually pretend like you're placing a suture almost at the base of the intestine and ligating so that it's not bleeding. And it actually is like a very prominent artery that'll spurt at you. And so they can be a pain. So I find trying to create an environment for success before you cut the intestine is important.
So I think surgery is very much an iterative, cumulative acquisition of skills. And when you begin surgery, you don't know how to hold a pair of needle drivers and you don't know how to tie a knot. I mean, it's very appropriate that you learn to do that on a silicone pad. And then the next challenge after that becomes how to adjust that suture tension appropriately to the tissue that it's being placed in. And then a silicone pad no longer works and you need something that simulates tissue. And then the next challenge after that becomes how do I do those things while working down and inside with another piece of bowel, trying to flop on top of the piece of bowel I'm working on. And then to learn that skill set, you need an actual full dog. And then the final part of it becomes how do I learn to do that with a patient that's breathing and moving and bleeding, where I feel the pressure of emotional responsibility to the patient and the owner to perform optimally. And for that, you then need a live patient. And you would never think that your entire surgical training could be accomplished on a silicone pad, but you would never begin with the live patient. For any skill set, there is a learning curve that exists. When you're low on that learning curve, you will have an error rate. And what is important is for that error rate not be transmitted to the patient. Learning in an environment, just in the same way that airline pilots learn in simulators, surgeons need that same opportunity.
So we've designed this so that you have repeatability. The organs that are interfaced are replaceable. Going further back, you can appreciate the stomach and the omentum, which is draping all of the intestine. This is a female dog. It's also designed as a spay trainer. We do have the reproductive tract represented. It also has the suspensory ligaments, the broad ligament, it has the horns, ovary. You would have to approach the abdomen, go in and break down that suspensory ligament, ligate the ovarian vasculature, and then come down, break it down and remove this as you would a common spay.
I think it's important to recognize that all surgeons, no matter how experienced and how proficient, will run into situations where either they've made an error or just the nature of the procedure that they're working on means that some untoward event begins to occur. Typically, that would be active hemorrhage. It might be loss of control of a ligature. It might be an insecure knot. It might be inadvertently traumatizing crucial tissue. And it's important, in my opinion, that students experience that emotion for the first time in a setting where there is not a live patient at risk and where there is someone available to mentor them through how specifically to address that issue. And so that's a great opportunity to give them their first introduction to that emotion and how to handle it, to perform competently under stress.
We're going to set up a clinical scenario where one of their pedicles secondary to a spay that they've been doing is now bleeding. The patient is starting to decompensate. Their anesthesiologist is panicking in a certain way and kind of set up a high pressure situation for them to try and figure out where the bleeding is and to then stop that bleeding. Because your blood or your heart rate has now come up to 160 beats per minute.
There must be something bleeding. Okay, I'm gonna check here. Oh, there it is.
And your patient's heart rate is 180 beats per minute.
And this stupid hemostat is not working.
We're up to 200 beats per minute.
Okay, bleeding is under control.
And our heart rate is coming back down slowly here. Once you're done, cut that and release the hemostat and see if it's bleeding.
Not bleeding. We saved him. We saved him.
And so I would say that was really good. You guys identified the major bleeding pedicle. The only thing that I would say, instead of going directly in with a hemostat, right, start with the Balfours, right, get in, improve your visualization, right? How did you feel as the crisis developed?
I mean, yeah, I do feel like the machine really hyped it up. Made me think something was really bad. But yeah, it was well controlled really quickly, and we were able to visualize everything really quickly. So
I think really the Sindava Lab accomplishes multiple facets or multiple goals. One is to try and remove students' natural fear of surgery. It's very hard to behave with good competency and calmness when you feel stressed or afraid. The second is to encourage an enthusiasm for the procedures and for surgery in general, and encourage them in the idea that your surgical training is an iterative process. You acquire a skill, you practice it, you discover that you need another skill, you go learn it, you practice that, you constantly improve, and that's a lifelong career commitment. And the third is to give them a basic skill set which will translate into any surgical procedure that they undertake.
Well, the most difficult thing about getting something like the synthetic canine into practice is the cost. We have a number of ways we can do to help universities and different places acquire them. It's an investment. It's definitely an investment. You know, the argument that we make, and the argument that's 100% true, is that you get superior training. You know, if you really want to be in the business of graduating students that are going to be able to practice and perform surgery, you're doing them a disservice if you don't, invest in this sort of platform.
For me, performing that kind of surgical training on a live animal which is being used solely for the purposes of training and is then euthanized is unacceptable for a large number of reasons, not the least of which that I believe it inculcates in the beginning surgeon the idea that the patient is disposable. I would not be ever willing to participate in a lab like that.
The only use of live animals in the training of surgery at the University of Florida is in our small animal hospital, where we have board certified surgeons who are doing clinical surgeries with our professional veterinary students, either assisting or watching. I am not a believer in terminal surgery. They don't do it in human surgical training, and I don't, it's my opinion that it's not something that we need to do in veterinary, and I've always felt that way.
You can learn good medicine and be ethical at the same time. And I believe in the long run, that will translate to being a better, more empathic doctor too, because you'll carry that burden. When I graduated, I did have to experience terminal surgery. In hindsight, I don't feel that that really taught me anything that helped me in the future. In fact, I still carry that scar. It all circles back to that core necessity on several fronts. One, to preserve our own emotional and ethical kind of compass, as well as to allow us to be better clinicians, to give our clients better service, better medicine. I love caring for animals. I see myself more as a pediatrician than a veterinarian. I see veterinary medicine and the practice of veterinary medicine more about preserving the bond. I feel a great weight of responsibility that the connection between that client and that patient, I mean, that's just pure love. People are appreciating this unconditional love that these companions give. It is really, it's a joy for me, actually.
Okay, so I'm hoping you can still hear me. I'm going to go back to my PowerPoint. I hope you enjoyed the opportunity to see the film and get a taste of what some of the greatest and most advanced methods are. It seems I have jumped out of my PowerPoint. I've just got to get back to the right position. Here we are. And while I appreciate that that is a particularly expensive tool, that doesn't mean it's completely unaffordable. They do last decades, those models, even if some of the organs need replacing. Oops, sorry. Here we are. At the same time, there are free and inexpensive tools that can be used instead or in combination with the more high fidelity and expensive models. So I think that's important to bear in mind what can be produced locally. So finishing off the types of alternatives and the advantages and the resources available, I'll now do in a fairly speedy way, because I'm aware we don't have so much time. When you do need hands-on experience of animals and animal tissues, we have really two areas: the ethically sourced cadavers and then the work with patients. The cadavers are, I think, a very important opportunity. You can get a body donation program for animal cadavers, and this program here, you can see the advertisement for on the right, is a national level program for collecting animals from private clinics. So it's a very, very good program of producing and supplying animal cadavers for chilling, freezing, preservation, and others. Preservation, of course, can involve plastination or other methods like the El Nadi technique, produced by Dr. Fawzi El Nadi, which is an inexpensive, low-cost, acetone-based way of preserving specimens for both anatomy, pathology, but also clinical skills and surgery. You can add perfusion and pulsate, and that's very good as well for creating a more live clinical and surgical situation, such as these models produced in Brazil. This can even be done in neurosurgery. That's Abud's method. You can add such perfused organs into tools like this POP trainer, the pulsating organ perfusion trainer from Optimus, the company in Austria, for laparoscopic training. And work with patients, as I mentioned, is another very important thing. In human medicine, you have the different levels and specializations, but also in veterinary medicine, where students need to really gain most of their skills within their degree. I think there's an important area there for spay-neuter work, working with organizations in order to get a lot of clinical and surgical experience under supervision, following on from the non-animal methods. And what's valuable about this, of course, is that it's case-based. It's highly realistic because it's with real patients, and it gives you an opportunity to have experiences right across from the very beginning of meeting the client to seeing the recovery with the operation in the middle. So work with clinics, a distributed system, as well as having teaching hospitals, all of that I think is very, very important and gives a lot of great hands-on experience to the students and the trainees. It also gives you the chance to learn caring as a clinical skill. And this, I think, is valuable for veterinarians, but also for doctors. You really get the chance to develop this when you use alternatives, including and especially the teaching hospitals. Animal volunteers is also another way of working with living animals, a chance for seeing living anatomy, to understand biodynamics, such as body painting of horses, watching dogs, companion animal dogs, and how they walk, what they look at. All of this can be considered work with animal volunteers if the animals only get rewards for that interaction. And as soon as they evidence any discomfort, of course, the class should stop. You can do experiments on yourself, as I mentioned, nerve conduction velocity experiments, a simple minor electric shock to the arm, looking at the nerve response. And I think there's a lot of apparatus where you can do such experiments on yourself. And that's a lot of fun, I think, good for medical students, but also for veterinary students and biology students as well. And in combination with software, really excellent for understanding physiology and pharmacology. You can do work in vitro, very good preparation for the growing areas within research and testing. This is also a potential replacement. And finally, ethical field studies, getting out of the lab into the field. Very good for biologists, but also some degree of veterinary student training as well can be done with looking at wild animals, stray animals, animals in the city. And this is very good, I think, in terms of the observation potential and seeing animals within their ecological context. So this brings us to another myth. Hopefully I've disabused this already, but the myth is that using alternatives means students won't have hands-on experience of animals and animal tissues. We have to ask, is it relevant? Do we really want medical students who know all about frogs and dogs. No, we want to look at the relevant reference object, human bodies, but also many other students won't be going on into their fields as a career. So we have to ask, is experience of animals hands-on relevant for many students? But of course, even then, replacement can involve animals, ethically sourced cadavers, and clinical learning opportunities, not animal experiments. Hands-on work doesn't mean animal experiment. And students, professionals will have continuing access with animals as the clinicians as well. So this is also extended hands-on experience and work with real animals over time. So the next part, and I need to wrap up very soon, is a few reflections on teaching objectives and the broader impact of alternatives. I think we can meet standard teaching objectives more effectively. Oh, I'm sorry, I could hear another voice, but did someone say something? Okay, I'll just wrap up. Standard teaching objectives can be met better using alternatives. The published studies demonstrate this. But also we can meet new objectives provided by the tools and the design. New surgeries, manageable only on the mannequins. Caring as a clinical skill. And you can also overcome what's called the hidden curriculum, the negative lessons that an animal experiment might teach you, such as teaching disrespect for animal life. Life science philosophy is about the study of life for biology, medicine as healing arts and sciences, critical thinking as an essential skill, and again, alternatives can help in this. Similarly with emotional literacy, that's awareness and skills, such as compassion and empathy, and ethical literacy as well, something that's very important within the professions, can be further defended and developed when using alternatives. Education can be more accessible and inclusive if you're not discriminating against students who might be uncomfortable or against harmful animal use. And so there's a civil liberties issue and a freedom of conscience issue. You can have a better reputation when you innovate and think about curricular change and improvement. It's very good for teachers and universities to think about developing their courses and enhancing them, giving a greater reputation. The animals, of course, are free from harm. The ecological impact can be reduced from the negative, or you can occasionally even have a positive impact with a good fieldwork project, for example. And the broader impact in terms of healthcare for humans and animals can also be enhanced as well. So the broader impact in society is good. Many alternatives are costly, but others are completely free. Free software, free body donation programs, and very inexpensive software and models, including ones you can develop yourself. There's also an opportunity to develop and sell alternatives as a teacher or institute. So I think overall the economic benefits are great. And legislation and regulations usually state that alternatives should be used wherever possible. And it's been great to see the very positive, progressive change at national level in India and elsewhere concerning zoology dissections, but also other areas like pharmacy too, where animal experiments have been stopped or the alternatives have been recommended. So that's a very progressive change, I think, happening at a national level in India. And I'm very happy we were able to contribute in some way to that process ourselves. So briefly, the resources that are available to support the process of replacement. We have our book From Guinea Pig to Computer Mouse, the documentary film you've seen one episode of, for which other episodes are under production, and a case studies book for next year. We have our website with free access databases, our alternatives database, a studies database, and soon the video database. You can search for alternatives, find out information about individual products and where to get them from. You can search for references and abstracts of, again, over 1,000 studies relating to the development and implementation of these tools, as well as comparative studies. You can borrow items from us. We have small grants and support the development of freeware. We support students who conscientiously object, and we do conferences, outreach tours, and training as well. There's also other items available, such as the virtual physiology programs that are free for students, even though faculties need to buy them, and other items you can contact me about. You can search through other bodies and journals like Intl and Altex for more information. Think about collaborating with your colleagues and across disciplines, because there's lots of great ideas and skills around for the production of new tools. And there's another couple of weeks left for applying for the Lush Prize, which rewards activity of replacement and innovation retrospectively every 2 years. And there's 2 more weeks to apply for this at lushprize.org. So in conclusion, I would say that alternatives are superior to harmful animal use in every respect, that 100% replacement is indeed possible, that there's a diverse range of tools and approaches with alternatives for every practical class, and that there are growing momentum and successes happening across the world with alternatives now seen as the norm at many, many different departments. So it's a win-win solution, multiple positive impact with including sustainable development more generally. Students benefit by having access to better teaching tools and approaches. Teachers can benefit because they can pass on their knowledge and skills more effectively. Society benefits as better trained individuals enter into the professions. And of course, the animals benefit as well. So very positive outlook, I believe. There's progress in many different countries, including in India. And do contact me for any further information. And I look forward to your questions in the few minutes we have left shortly. You can contact me here. Thank you very much.
Thank you very much, Prof. Nick, for this highly elaborative and comprehensive presentation with us, and also very interesting movie. Now, if there is any comments and questions, you are welcome.
Thank you, sir, for this wonderful session. Now I request Dr. Farhan Ahmad Khan, sir, to hand over the memento to Dr. Misbauddin, sir.
Thank you for this wonderful session, sir.
Thank you very much. Great to be at the event, and I wish you a good final few sessions and conclusion.
I'm delighted to announce a brief interlude in our proceedings, a well-deserved tea break, which will be followed by a valedictory function. I request everyone to move to the designated refreshment area, and the tea break will be of 10 minutes. Thank you.
Assalamu alaikum and very good evening and very good evening to everyone present here. I, Dr. Anam,
I, Dr. Abhinav Tiwari,
Junior Resident from Department of Pharmacology, JNMCH, AMU, welcome you all. Today marks the culmination of a thought-provoking and enlightening journey, the valedictory function of our International Conference on Alternative to Animal Experiment. We have had the privilege of hosting and learning from distinguished guests, eminent speakers, and zealous researchers over the past few days, contributing to a collective effort that will undoubtedly shape to reform the future of scientific research. With great honor, we welcome distinguished individuals to grace the stage for the closing ceremony. Honorable Chief Guest Professor Abdul Alim Sir, Dean, Student Welfare, AMU. Professor Y.K. Gupta Sir, esteemed President of the Society of Alternatives to Animal Experiments, India.
I request everyone sitting in the audience to come forward, please, to take the front seats.
Professor M.A. Akbar Shah Sir, General Secretary of Society for Alternative to Animal Experiments, India.
Professor Dr. Syed Ziraur Rahman Sir, Organizing Secretary, Chairman, Department of Pharmacology, JNMC. Yes, sir. Mr. Puran Chand Sir, Assistant Commissioner of Drug, Aligarh Division, and current National General Secretary of All India Drug Control Officer Welfare Organization. And lastly, Dr. Irfan Ahmed Khan Sir, Assistant Professor, JNMCH, AMU.
We appreciate your support and encouragement throughout this conference. In our pursuit of knowledge and ethical practices, we are privileged to have Dr. Shamir P.S., Junior Resident of Department of Pharmacology, here with us to recite the few verses from the Holy Quran. We are privileged to have Dr. Shamir Pierce, Junior Resident of Department of Pharmacology, here with us to recite the few verses from the Holy Quran.
Assalamu alaikum wa rahmatullah. أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. هو الله الذي لا إله إلا هو عالم الغيب والشهادة هو الرحمن الرحيم. هو الله الذي لا إله إلا هو الملك القدوس السلام المؤمن المهيمن العزيز الجبار المتكبر سبحان الله عما يشركون. هو الله الخالق البارئ المصور له الأسماء الحسنى يسبح له ما في السماوات والأرض وهو العزيز الحكيم. صدق الله العظيم.
Thank you, Shamir. It is our distinct pleasure to request Mr. Puran Chand sir to confer a memento as token of appreciation to Professor Abdul Aleem sir. Yes, sir. Mr. Puran Chand sir to Professor Abdul Aleem sir. Now we kindly request Professor Syed Ziaur Rahman sir to present a memento to Mr. Puran Chand sir.
Now we will have the honor of hearing from Professor Dr. Syed Ziaur Rahman sir, Chairman, Department of Pharmacology, JNMC AMU. I humbly request sir to please take the stage and share his valuable views for the conference with us.
It's not my valuable views, but I am here just to thank everybody. So first of all, I would like to welcome our chief guest, Professor Abdul Aleem, sir, Dean Student Welfare, Aligarh Muslim City, Professor Y.K. Gupta, President, the Society for Alternatives to Animal Experimentation, the guru of all the pharmacologists, Dr. Puran Chand, the Assistant Commissioner, Drugs, Aligarh Division, Professor Raisuddin Sahab, the General Secretary of the Society, Dr. Irfan Ahmed Khan, Assistant Professor, Department of Pharmacology, Dr. Anika Pandey, Dr. Savita, Dr. Pratiksha, Professor Shams Barayuni Sahab, Zawar Rahman Siddiqui Sahab, Anwar Abdul Rauf Sahab, Chairman, Department of Involvia. I can't forget Raisuddin Sahab, Sheikh Raisuddin Sahab, the Vice President, the main supporter of the society, the Metic Prize winners, both oral and poster presentation. Ms. Baudin from Saudi Arabia, Dr. Ahmed Najmi from Bhopal, all the residents, the teaching staff, and the non-teaching staff of the Department of Pharmacology. So just to thank you all, and it's a 2 days hectic conference. If there's any shortcoming, I take the responsibility, because this is something which I took the responsibility for the society, and the society has given me the responsibility to organize this conference in Aligarh Muslim City. So it was a challenge, but still we tried our best. But I know that nothing is foolproof, and it's like we try our best, but there are sometimes very shortcomings. In fact, what I observe is very poor attendance, and we should have included our undergraduate students also. But these students are busy in their terminal examination. Even today they have terminal examination, microbiology terminal examination. Tomorrow they'll have the pharmacology terminal examination. So in this valedictory function, we are supposed to announce the Metic Prize, which was donated by Christian Velivoisen, which our General Secretary, Dr. Akbar Shah, will announce the winners, like who's the first, second, third, and prize winners. So on my behalf, I welcome once again our chief guest and the guest of honor, Dr. Puran Chand. Dr. Puran Chand is very busy person. He's looking after the Aligarh division, and so many things he has to take the responsibility. So I'm really lucky that he has joined in our very first inaugural session, and he chaired a lecture given by Professor Y.K. Gupta, and once again he is here. So it's your dedication towards the society that you are here. I'm really thankful, and I'm so thankful to the chief guest, Professor Abdul Aleem Sahab. Again, Dean Student Welfare is also very responsibility, you can say. So although Dr. Irfan Ahmed Khan will give the formal vote of thanks, but on my behalf, I once again welcome you, and thanks all the workers, volunteers, and the students, and everybody who has cooperated us. Thank you so much.
The dedication and hard work of our society members have been the backbone of this event. I humbly request the General Secretary of Society for Alternatives to Animal Experiments India, Professor Dr. M.A. Akbar Shah sir, to share his views on the outcomes and discussions of this conference. Sir.
So, Honorable Chief Guest of this function, the valedictory function, Professor Abdul Aleem, the Dean of Student Welfare, Chief Guest of Honor, Dr. Puran Chand, my President, Dr. Gupta, my Vice President, Dr. Raisuddin, the Organizing Secretary, Dr. Ziaur Rahman, and Dr. Irfan, the invitees, VIPs, and all my members of the society, their executive committee members who have been supporting me in conducting this conference, along with Dr. Ziaur Rahman and my dear students and other participants. I'm immensely pleased to be sharing the dais today with during the valedictory function. I just want to comment that the organizing of this conference has been really exemplary. Our Rahman has taken up the responsibility. He was not getting much fund, and under the restraints of fund limitation, he has done his best to provide all the facility, all the support that required to conduct this conference in the manner exemplary. So my compliment to Dr. Ziaur Rahman and his colleagues in the department, starting from the professors, the assistant professors, associate professors, the technical assistants, and other laboratory supporting staff, all the students, when they were working as like different parts of one body, right? It's a very homogeneously knit organization. And I could see Ziaur Rahman delegating responsibility to his colleagues and students, and I see them wonderfully transforming and translating his thoughts into action. So my compliment and grateful thanks to all the colleagues and all the students of the department. Well, I have one or two things briefly to tell you. My vice president has been telling that you should tell one or two points about the society to the audience. So I make this opportunity to tell you that this Society for Alternatives to Animal Experiments is essentially meant to, as an academic venture, of enlightening and sensitizing the stakeholders, the academia, the research scientists, the regulatory people, of the problems in using animals in experiments, particularly concerning concerning the meant to be submitted to regulatory approval. I used to say the two major branches of science, toxicology and biomedical science. So these are the two areas where we use great many number of animals, and there have been a lot of criticism, a lot of problems using these animals in the research and the outcomes. Finally, these are meant to be made for whether the chemicals are good for health or not good for health. The drug is really translated into therapy or not. So these are very important areas, and where animal experimentation is not definitely known to be giving the real required result, and it could lead to uncertainties in terms of prescription of drugs and the practice of the chemicals in the food, pharmaceuticals, and so on. So we have been, this has been a big demand at the global level, and in India, this movement was started somewhere around 2000, and with these 20 years, many things have happened, and we had a Center for Alternatives in India at Bharatdasan University, which I was able to mobilize with money from Doran Gumspin Foundation in Switzerland. And then we had a National Center for Alternatives. My center was taken over as a National Center for Alternatives with funding that came from the University Grants Commission. And subsequently, I mean, it remained to be, it happened that it is limited to a small area in a small place. So we thought we should expand it further. So to make it a national endeavor, we thought, some of us like-minded people, senior professors, we thought it fit to establish a Society for Alternatives, and that was done in 2018. Society for Alternatives to Animal Experiments, India. That's the name of the society. We launched it in 2018, and very pleasantly, the first enrollment was that of Dr. Ziaur, the first person to become a member of the society. Even I did not. Even before I became a member, Raizuddin became a member. Ziaur Rahman was my friend for 20 years. So she was the first to become a member of this society, and it has grown from step to step, leaps and bounds. So now, in 2018, we had the first conference in Jamia Hamdard in New Delhi under the organizing secretaryship of Professor Raizuddin. And 2019, we had the conference in Mumbai. NFB, National Facility for Biopharmaceuticals, they were kind enough to host it. And 2021, we were not able to conduct it because of the COVID. And we had online, of course, we had online conferences. 2022, it was one of the very extravaganza that happened in the University of Kerala, Thiruvananthapuram. Very beautifully conducted conference. And then 2023, we have it in Aligarh Muslim University, Department of Pharmacology. A brief history to track, starting 2018 to 2023. Even at the times of crisis, we were able to conduct the conference, so we are up to date. And we have been participating in not only education, we are also in motivating and sensitizing research community, the regulatory community, about the need to change from animal experimentation to more relevant and more appropriate alternative methods. So we have been doing it for 6 years, and we hope to grow to further strength. As of now, we have 64 members, and we look forward to cross the 100 in maybe another few months with the help of all my friends who are already in the society. So we have been doing the best that is possible to tell people about the problems in the animal experimentation and the outcomes and the inferences that are there from there. And we have done our best, and we hope to continue to do in the days to come. And we look forward to associate with the Aligarh Muslim University for many more activities in the years to come. I am associated with this many years, right? For several contexts and several programs, several activities, research, so on. So I look forward to come back to Aligarh Muslim University for many more endeavors as regards the Society for Alternatives to Animal Experiments. And the students and the teaching community, they can look forward to me and my colleagues like Sureshuddin Saab or Gupta Saab or Pradiksha, Angida. You can seek our help in any context of alternatives and our own academic streams. We have our own specializations, and you can reach us for any type of support that you would require from organizational point of view and from educational or academic point of view. Having said this, I would like to once again thank Ziaur Rahman and his wonderful team for the magnificent work they have done, and we wish them to have many more such events in the years to come. Thank you so much.
Thank you, sir, for motivating us. Now I kindly request Professor Y.K. Gupta, sir, esteemed President of the Society of Alternatives to Animal Experiment India, to share his views on the success of this conference.
आदरणीय प्रोफेसर अब्दुल अलीम साहब, डीन स्टूडेंट वेलफेयर, गेस्ट ऑफ ऑनर और मेरे बड़े अच्छे दोस्त बन गए हैं डॉक्टर पूरन चंद जी, असिस्टेंट ड्रग कंट्रोलर, ड्रग कमिश्नर अलीगढ़ जोन, डॉक्टर जियाउर रहमान और डॉक्टर इरफान खान, और ऑफ कोर्स द जनरल सेक्रेटरी एंड जिसे हिंदी में हम कहते हैं सूत्रधार ऑफ दिस कॉन्फ्रेंस, Dr. Akbar Shah and Dr. Raisuddin Saab, and three pillar ladies, Ankita, Savita and Pratiksha, the three young beautiful ladies who are very active right from the beginning, and they are pillars, if I say. Ladies and gentlemen, and all the faculty from this institute, Aligarh University, and अभी मैं एक चीज देख रहा था, दोपहर को लंच में जब हम खाने गए तो मेरे पास पीछे पीछे दो कुत्ते आए, वो वर वेजिंग पूंछ हिला रहे थे ऐसे ऐसे, तो मुझे लगा कि ये हमको थैंक्स दे रहे हैं 2 दिन मीटिंग करने के बाद में. यू हैव टॉक्ड अबाउट लॉट ऑफ लाइफ सेविंग एफर्ट्स फॉर अस. तो फिर मेरे रास्ते में सोचा ये तो बेचारे पूंछ हिलाते ही रहते हैं. That means they are always so grateful to us, but we have never been so grateful to them. We never loved them, though we love them when it is pet of ours. हमारे घर में जब हमारा कुत्ता होता है तो हमारे कुत्ता हमारा बिस्तर पे सोता है, isn't it? लेकिन जब सड़क पे कुत्ता होता है तो हम लात मार के उसको पत्थर मार देते हैं, लेकिन फिर भी वो पूंछ हिलाता है. I think that is what the innocence of these I think animals. So I said in the yesterday also कि ये जो एनिमल्स हैं, चाहे चूहा हो, चाहे कुत्ता हो, चाहे बंदर हो, they cannot express their anger, they cannot express their feeling, but it is our duty to understand their feeling. और हम ये नहीं कहते कि हम आदमी की जान बचाने के लिए चूहे की जान को न लें, हम लेंगे, क्योंकि ये हमारा काम है, क्योंकि आदमी की जान बचाना हमारा पहला काम है। लेकिन अगर हम आदमी की जान बचाने के लिए दूसरे की जान न ले सकें तो इससे अच्छा कुछ नहीं हो सकता। एंड दैट इज द पर्पस ऑफ दिस सोसाइटी। तो हम हमेशा यही कहते हैं कि हम झंडा लेके हम नारेबाजी नहीं करेंगे कि एनिमल एक्सपेरिमेंट जो कर रहा है उसको अरेस्ट करो, उसकी लैब बंद करो, नहीं। लेकिन जो एनिमल एक्सपेरिमेंट कर रहा है और उसका ऑल्टरनेटिव हो सकता है, उसके बाद भी जो कर रहा है, तो हमें झंडा लेना है। दैट इज व्हाट दिस सोसाइटी इज फॉर। एंड हमारा ये जो 2 दिन का था, इसके बाद में आई थिंक पीपल मस्ट हैव रियलाइज्ड आफ्टर listening to the talks that there are so many development which have taken place, so much that maybe after 5 or 10 years we will be at a different forum, जहाँ पे कि ये जो हमारे जितने इन विट्रो हैं, जितने ऑर्गन चिप्स हमारे हैं, ऑर्गेनॉइड्स बनाने वाले हैं, IIT चेन्नई और IIT रुड़की, डिफरेंट इंस्टीट्यूशंस जितना इस पे काम कर रहे हैं, जो हमें सिर्फ एक ही जरूरत है, हमारा लक्ष्य है कि ये जो इन्होंने डेवलप किए हैं ऑल्टरनेटिव्स, इनको हम वैलिडेट करके, वैलिडेट करने के बाद में हम इसको रेगुलेटरी स्टेटस में ले आए, एंड देन दे विल बी यूजफुल। जब तक हम उसको रेगुलेटरी अप्रूवल नहीं कराएंगे, तब तक हमें एक डाइकोटमी, हम कह रहे हैं अल्टरनेट एनिमल्स यूज करो, रेगुलर कहता है नहीं आप तो चूहे और बिल्ली और कुत्ते में डेटा लेके आओ। तो इंडस्ट्री क्या करेगी? इंडस्ट्री विल नॉट वेट फॉर कि आप ऑल्टरनेट डेवलप करो, तब तक हम अपने मॉलिक्यूल को हम ड्रग कंट्रोलर को सबमिट नहीं करेंगे। दे आर गोइंग अब्रॉड, दे आर गोइंग टू चाइना, दे आर गोइंग टू कोरिया, दे आर गोइंग टू इंडोनेशिया फॉर गेटिंग द रिजल्ट्स डन इन डॉग और मंकीज, बिकॉज हमारे यहाँ फैसिलिटी बंद हो गई है, नहीं है। सो हमारा ऑब्जेक्टिव ये है कि हम इन ऑल्टरनेटिव को ला करके रेगुलेटर से अप्रूव कराने तक we have to make an effort, and that is what the purpose of this society is. And yesterday we decided that we will meet regulator more frequently, maybe every 3 and 6 months, giving the bundle of the evidences that ye hamare paas evidences hain, whether you accept this or not, so that they are also mindset will change. तो ये बहुत जरूरी है, और इसके लिए मल्टीडिसिप्लिनरी अप्रोच होना जरूरी है। आई एम वेरी हैप्पी दैट डीन स्टूडेंट वेलफेयर इज विद अस, क्योंकि डीन स्टूडेंट वेलफेयर विल कम्युनिकेट थ्रू his message to the all students, because ultimately later on these students will become the scientists and will become the policy makers. And Aligarh University is known to create policy makers. Aligarh University is a hub of culture and education. We are proud of Aligarh University, and we are, और अलीगढ़ यूनिवर्सिटी में आके वैसे ही यहाँ पे लगता है कि हम थोड़ा ज्ञान बढ़ गया हमारे, we have become richer in education. So I think for organizing this, I say Dr. Rahman is a courageous person, and courageous person because इन्होंने last time risk ली कि छोटी सी सोसाइटी है, इस सोसाइटी को कोई पैसा वैसा देता नहीं है as compared to cardiovascular society or diabetes society where people are eager to give money and sponsor it. Here there is no money because there is hardly any sponsor. Maybe after 10 years there will be, but he took a chance. He is a courageous person. तो लीडर करेजियस तभी होता है जब उसके पीछे एक फौज लड़ने के लिए तैयार है। तो इनकी ताली कम, लेकिन इनकी फौज के लिए हम ताली ज्यादा बजाना चाहेंगे। द वॉलंटियर्स, द रेजिडेंट्स, and I was feeling that I am a VIP in this meeting, but when I realized everybody is VIP. किसी को ये फील ही नहीं हुआ यहाँ पे आने के बाद में, चाहे वो रईसुद्दीन साहब हो, चाहे वो अंकिता, प्रतीक्षा, सविता, ये लोग हों, वो ये 3 ट्रायो हैं, एक ही जगह बैठे रहते हैं, गप मारते रहते हैं। So and this is something great that you make everybody so comfortable. दोपहर को जलेबी, शाम को पोहा, और उसके बाद में आई थिंक हमने सुना ही था कि अलीगढ़ की खातिरदारी बड़ी फेमस होती है, लेकिन अब हमने महसूस भी कर लिया और हमने उसका आनंद भी ले लिया। सो हमारे लिए आपको सबको बहुत बहुत धन्यवाद। मैंने देखा है बहुत सारे वैलिडिटरी फंक्शन मैंने अटेंड किए, और अक्सर जिस कॉन्फ्रेंस में 4000 5000 की अटेंडेंस होती है, वैलिडिटरी के दिन सिर्फ डायस पे होते हैं और 2-3 आगे लोग होते हैं, क्योंकि उनके साथ में वॉलंटियर्स। इस कॉन्फ्रेंस में 100 रजिस्ट्रेशन और 150 लोग यहाँ पे बैठे हैं, 100 से ज्यादा बैठे हैं। आई थिंक दिस इज समथिंग एक्स्ट्रा ऑर्डिनरी, एक्स्ट्रा ऑर्डिनरी ऑर्गेनाइजेशन एंड एक्स्ट्रा ऑर्डिनरी लव एंड फ्रॉम ऑल ऑफ यू। ये मत सोचिएगा कि वैलिडिटरी एड्रेस में हॉल खाली है। इतना वैलिडिटरी एड्रेस, वैलिडिटरी फंक्शन के लिए ये हॉल, हम इसको कहेंगे खचाखच भरा हुआ है एज कंपेयर टू मोस्ट ऑफ द वैलिडिटरी। अगर आप, मैं आपसे कहूंगा कि अब आप वैलिडिटरी फंक्शन अटेंड किया करें, कॉन्फ्रेंस करो ना करो, वैलिडिटरी फंक्शन में जरूर जाके देखो, तो पता लग जाएगा कि ये वैलिडिटरी फंक्शन बाकी वैलिडिटरी फंक्शन से कितना सक्सेसफुल है। सो थैंक यू वेरी मच टू ऑल ऑफ यू जो यहाँ पे हैं, और आई थिंक दिस इज ऑल बिकॉज ऑफ डीन स्टूडेंट वेलफेयर, एंड मुझे लगता है कि आपका अट्रैक्शन है जो सब लोग यहाँ पे रुक गए। आई नेवर न्यू दैट द डीन and are so popular. So thank you very much. We will meet again in this wonderful city of hospitality and affection. और फिर मिलेंगे। धन्यवाद and thanks to all organizers.
Thank you so much, sir. Now, I humbly request Mr. Puran Chand, sir, Assistant Commissioner of Drug, Aligarh Division, to express his views regarding the conference.
सभी लोगों को मेरा नमस्कार। मंच पर बैठे विराजमान हमारे सीनियर्स, मैं सीनियर्स ही कहूँगा, काफ़ी बड़े हैं और काफ़ी सीनियर्स हैं मुझसे, और विशेष तौर से हैं प्रोफेसर वाई गुप्ता, जो कि वाकई इन्होंने कहा था कि एक शब्द इन्होंने कहा कि ये तो मेरे दोस्त बन गए। ये मेरे लिए गर्व की बात है, जबकि मुझसे काफ़ी सीनियर्स हैं, और ये फक्र की बात है दोस्त वर्ड कहना, ये बहुत बड़ी बात है। थैंक यू सर। देखिए, 2 दिन की कॉन्फ्रेंस में मैं बहुत थोड़ा कम समय ही कल यहाँ आया था, और मैं माफ़ी चाहूँगा, उसके बाद मैं थोड़ा सा दिन में 1 बजे बाद चला गया कुछ विशेष व्यवस्था के कारण। मैंने 2-3 लेक्चर सुने थे, 2-3 चीज़ें सुनी, क्योंकि मैं काफ़ी टाइम से इस फार्माकोलॉजी डिपार्टमेंट से कट था एडमिनिस्ट्रेशन में आने के बाद। कल मैं रिफ्रेश हुआ थोड़ा सा, कि अपने लोगों के बीच में आया, परिवार में आया। विशेष तौर से कल जो मैंने 2 थीम देखी, 2 क्या सभी अच्छी थी। विशेष तौर से वाई गुप्ता जी के तो मैं तारीफ करने से रुक ही नहीं सकता, मैं बिल्कुल किसी तरीके से। कल इंग्लैंड से आए प्रतिनिधि सविता, यस, कल इन्हों का जो एक अंदाज था बोलने का, एक्सीलेंट, एक्सीलेंट, इसके लिए मैं कहूंगा एक बार तालियां हों के लिए। और बाकी कल जिस तरीके से चल रहा था, मैं कॉन्फ्रेंस में 2-3 टॉपिक मैंने देखे। एक होता है क्लासरूम, और एक होता है कि कोचिंग। आजकल एक प्रचलन चला है ना कि बच्चा कहता है मैं कोचिंग गया, ट्यूशन गया। तो कल इस तरीके से समझाई जा रही थी चीजें, जैसे कि एक कोचिंग में, एक ट्यूशन पे एक टीचर बैठ के आराम से समझा रहा है, और क्वेश्चन आंसर भी। बहुत बड़ी बात है ये। और जैसा कि अभी 2 दिनों से आई बात समझ, ठीक है, हमें जानवरों पे किसी तरीके का कोई अत्याचार न हो। अच्छा, मैं हिंदी बोल रहा हूँ, कोई प्रॉब्लम तो नहीं है ना कहीं कोई? मैं थोड़ा सा क्या है कि एडमिनिस्ट्रेशन में आने के बाद बहुत कुछ कट हो गया हूँ मैं कुछ चीजों से, कोई ऐसी प्रॉब्लम तो नहीं, बस अपनी जुबान है, बस इसी कारण से। तो इसमें यह देख रहे हैं कि वाकई सही है कि हमें एक्सपेरिमेंट इस तरीके के करने चाहिए जहां जानवरों का उपयोग कम से कम हो। मैं कल एक थीम पढ़ रहा था, एक जानवर की तरफ से ही है, जानवरों को अपने जीने का अधिकार है, और हमें सिर्फ इसलिए उन पर एक्सपेरिमेंट नहीं करना चाहिए क्योंकि हम ऐसा कर सकते हैं। वे हमारे अच्छे साथी बनने को तैयार है, ये निश्चित मानिए आप, वो हमारे अच्छे साथी हैं। ठीक है, ये मजबूरी है हम लोगों की कि एक्सपेरिमेंट भी करने जरूरी है और मानव हित के लिए, मानव सेवा के लिए वो भी जरूरी है। विकल्प ढूंढे जा रहे हैं, जैसा कि मैं रिसर्च देख रहा हूँ कि करीब 400 ऐसे तरीके निकाले गए हैं वर्ल्ड के अंदर जहां अल्टरनेट किया जा सकता है, कहीं ना कहीं अल्टरनेट किया जा सकता है। मैं 2 दिनों से यही दोबारा से रिफ्रेश हो रहा था गूगल वगैरह पे भी, और सरिता जी का भी मैंने गूगल पे सर्च किया था, और उस पे काफी कुछ मैंने उसमें से भी टॉपिक्स देखे। बहुत अच्छे टॉपिक्स हैं आपके उसमें, बहुत अच्छे आपके कमेंट्स हैं, और बहुत ज्यादा आपने उसमें लिखा है। और मैं यही कहूंगा कि भाई मुझे यहाँ पर मौका दिया अपने लोगों के बीच में, परिवार में मिलने का, उसके लिए धन्यवाद। और 2 दिनों से मैं देख रहा हूँ कि जैसे होता है ना किसी मैच के मैन ऑफ द मैच किसको दिया जाए, यहाँ सभी मैन ऑफ द मैच, सभी, कोई एक नहीं है, सभी अपने अपने स्तर पे अपना अपना काम कर रहे हैं। स्टूडेंट्स हैं वो भी देख यहाँ मौजूद हैं, सीनियर्स जो कि रिटायर हो गए, कल मैं नोट कर रहा था डॉक्टर सिंघल जी कल आए, मतलब एक सीनियर मोस्ट पर्सन आए, कॉन्फ्रेंस में आए, ये एक बहुत बड़ी बात है। ऑर्गेनाइजिंग सेक्रेटरी रहमान जी तो मैं देख रहा हूँ इस तरीके से लगे हुए हैं जैसे कोई होता है ना कि एक बेटी की शादी होती है तो वो बेटी का पिता पूरे सारे कामों को करने लगता है, और वो इस चीज का कर रहे हैं अब बारात विदा हो जाए किसी तरीके से। तो तो सर मैंने कोई कल्पना गलत तो नहीं की है इस बात की, ऐसा महसूस कर रहे होंगे आप शायद। हाँ जी। जी सर। तो अगेन धन्यवाद, नमस्कार सभी लोगों को। और हाँ, किसी भी स्तर से अगर इस किसी भी सोसाइटी को कहीं गवर्नमेंट स्तर से, कहीं मेरे स्तर से, कोई फार्मा डिपार्टमेंट से, कहीं भी कोई भी ड्रग डिपार्टमेंट से कोई भी सहयोग की जरूरत हो, आपका एज ए स्टूडेंट मैं हरदम साथ हूँ। थैंक यू।
Thank you, sir. We are truly honored to have Professor Abdul Aleem sir with us. Sir, I would request you to share your views regarding the conference with us.
Good evening. Assalamu alaikum. Mr. Puran Chandji, Assistant Commissioner, Drug Legal Division. Professor Y.K. Gupta, President of the Society. Professor Akbar Shah, General Secretary of the Society. Professor Sayyid Ziaur Rahman, Chairman, Department of Pharmacology and Organizing Secretary, International Conference. Dr. Irfan Ahmed Khan. Professor Raisuddin Sahab, Vice President of the Society. Dr. Shams Badayuni, प्रोफेसर ज़ियाउर रहमान सिद्दीकी, डिप्टी DSW AMU अलीगढ़, प्रोफेसर अनवर शहजाद और मेरे दोस्त पास में ही, और अभी 3 का नाम लिया गया जो सोसाइटी की पिलर हैं, सरिता, अंकिता, प्रतीक्षा, डिग्नेटरीज ऑफ द डाइस, पार्टिसिपेंट्स, all faculty members, dear students, ladies and gentlemen. This is my privilege and extend my thanks to the chairman and his team that you have invited me as a chief guest on this special occasion, on this international conference on alternative to animal experiment and 6th annual meeting of the society. What are the impact, importance, connectivity and relationship between science, society and culture? Where science is applicable in the society as to develop and maintain the culture, there is wide range of the discipline in the term of education, such as medicine and science, etc. Scientific and technological activities refer to the creation of the knowledge through the discovery of the new natural laws and principles and search for alternative to the animal experiment, and for this sufficient accumulation of scientific knowledge is required, which is strength of the theme of this conference. Many of the participants must have certainly benefited with this international conference. I congratulate for successful conduct of his of this conference and further extend my thanks to the chairman, Professor Sayyid Ziaur Rahman Sahab and his team, our organizer, for the successful completion and result-oriented conference. Thank you very much. ये अभी एक बात मुझे हिंदुस्तानी में कहना है, क्योंकि अभी जो न्यू एजुकेशन पॉलिसी 2020 आई है, मुझे लगता है उसकी अक्कासी प्रोफेसर वाई के गुप्ता ने की है। कि वाई के गुप्ता साहब ज़ाहिर है बड़े लर्नेड प्रोफेसर हैं, मगर जिस अंदाज में उन्होंने यहाँ पर अपने जज्बात की अक्कासी की है सोसाइटी के बारे में और अलीगढ़ यूनिवर्सिटी के बारे में, मैं उनका तहे दिल से शुक्रिया अदा करता हूँ कि आपने अलीगढ़ मुस्लिम यूनिवर्सिटी के बारे में जो जज्बात को इजहार किया है। और प्रोफेसर ज़ियाउर रहमान साहब जिनके बारे में आपने बताया है, ज़ाहिर है कुछ चीज़ें विरासत में मिलती हैं। अगर आप लोगों ने न देखा हो तो मैं आपसे रिक्वेस्ट करूँगा वाई के गुप्ता साहब, तो सर का जो कंट्रीब्यूशन है, जो वालिद मोहतरम है इनके, मुझे लगता है अलीगढ़ में कम लोगों का इतना बड़ा कंट्रीब्यूशन होगा, कितने पूरी कोठी को अपने उन्होंने म्यूजियम में तब्दील कर दिया है। और वही चीज़ मुझे लगता है कि वो डॉक्टर ज़ियाउर रहमान साहब को विरसे में मिली है, जो आपने बताया कल से आज तक इस तरह से लगे हुए हैं। मैं तो इन्हें स्टूडेंट लाइफ से जानता हूँ, जब ये MBBS के स्टूडेंट थे और हॉल मैगजीन के एडिटर थे। इनकी उस वक्त की मैगजीन प्रोफेसर KP सिंह साहब जिनको मेरे उस्ताद रहे हैं, उस वक्त इन्होंने मैगजीन निकाली थी। कहते हैं ना, आप बचपन से ही किसी के पालने में वो नजर आ जाता है। जब ये स्टूडेंट थे MA के, वो MBBS के, तब उन्होंने जो 2 मैगजीन निकाली थी अलीगढ़ के नंबर पर और वाइस चांसलर के बारे में और अलीगढ़ के बारे में, वो भी आज भी तारीखी हैसियत रखती हैं। तो मुझे ये लगता है कि सैयद प्रोफेसर ज़ियाउर रहमान साहब जिस तरफ बढ़ रहे हैं आगे, और मुझे लगता है अपने वालिद की तरह ही ये भी अपने सब्जेक्ट के अलावा, सब्जेक्ट देखिए हकीम साहब कितने बड़े अपने एक्सपर्ट हैं, मगर उसके अलावा उन्होंने जो सोसाइटी को दिया है, 50 बच्चे उनके यहाँ पढ़ रहे थे फ्री, उनकी मतलब उसमें रीडिंग रूम में। तो इस तरह से हम इस कॉन्फ्रेंस से या ऐसे लोगों से हम वो भी सीख सकते हैं कि ज़ियाउर रहमान साहब जैसे लोग सोसाइटी को क्या दे रहे हैं। जो अल्टरनेटिव अरेंजमेंट जो आपने कहा एनिमल्स का, आपने सही कहा, अगर अल्टरनेट कोई हो सकता है तो उसे हमें तलाश करना चाहिए। इसी तरह से जो लैंग्वेज है, अंग्रेजी में बात होनी चाहिए, जो अपने जज्बात की अक्कासी है वो हिंदुस्तानी में होती है। तो मैं अब इन्हीं अल्फाज के साथ प्रोफेसर सैयद ज़ियाउर रहमान साहब को मुबारकबाद देता हूँ और उनकी पूरी सोसाइटी को, जो इन्होंने एक सब्जेक्ट यहाँ पर रखा है और एक अल्टरनेट दे रहे हैं एक्सपेरिमेंट के लिए एनिमल को, बहुत ही इम्पोर्टेंट है। एक दिशा देना जिसे कहते हैं समाज को, मुझे लगता है एक दिशा मिलेगी। एक इस कॉन्फ्रेंस की कामयाबी के लिए आप सबको, आपकी पूरी टीम को, पूरे डिपार्टमेंट को और सोसाइटी को तहे दिल से मैं मुबारकबाद देता हूं और आप सबका शुक्रिया अदा करता हूं कि आपने मुझे बुलाया। बहुत बहुत शुक्रिया।
Let us proceed to a moment of recognition and celebration, the distribution of METEC Prize. We had the pleasure of acknowledging the best and the brightest among us. Now I would like to invite Professor Dr. M.A. Akbar Shah sir to come over the podium to announce the results of oral and poster presentation. Along with that, I would request I would also request our Honorable Chief Guest, Professor Abdul Aleem sir, to come forward to award the victorious participants.
So before I break the ice, I would like to make one or two announcements, and then I take over the announcement of the award winners. In my address, I forgot to tell you one important message, that the society is, in addition to what we have been conducting in India, is recognized. They are an MOU with the Japanese Society for Alternatives to Animal Experiments. We have MOU with the European Society for Toxicology, is USAT, and we are MOU with the American Society for Cellular and Computational Toxicology. That tells you that we are recognized at the global scale. And further to add to that, We are a constituent of the Asian Federation of Societies for Alternatives, and the member countries, member societies for this federation is the Japanese Society for Alternative Animal Experiments, the South Korean Society for Alternative Animal Experiments, and the Indian Society for Alternatives to Animal Experiments. So this federation, like the individual society, this federation also used to conduct the complex meetings with all constituent societies. We had a meeting of the Asian, that's called the Asian Federation, or the Asian Congress. We call it the Asian Congress. The Asia Congress, the first Congress was held in Japan in 2016. And the next meeting was held in 2021-22 at Jeju Island in South Korea. I attended both the meetings, had the pleasant privilege of participating in both the meetings. And the next meeting of the Asian Federation is presently being assigned to our society, the Indian Society for Alternative Animal Experiments. And we have identified the great institution in Delhi, the institution where we had the first meeting of the society, the Jamia Hamdard. So that will be the venue. They will be the host of this Asia Federation, Asia Congress, that will be held during November, December, the convenient time, 2024. Okay. So after this meeting, we will have the next meeting, which will be the Asia Congress meeting, where the annual meeting of our society, Indian Society, will also be held. It will be a kind of a sandwich program. I invite friends and colleagues students from Aligarh Muslim University and others who have come from other institutions also to make it possible to join us in our next meeting, the Asia Congress, to be held in Delhi in November, December 2024. So that's the announcement I thought I should make, and I forgot to tell you that. Can you have a list? Okay. So now I have come to break the ice, and the first to announce is the third prize for poster presentation. It goes in that order. And the third prize winner is Sabah Afshin from Jamia Hamdard. Is he or she there? Left. So will somebody receive it? Come. So her colleague from Jamia Hamdard is receiving it. Sabah Afshin from Jamia Hamdard. Chief guest. Oh. Yes. Thank you, sir. The second prize winner for poster is Ikra Subhan, Department of Zoology, Aligarh Muslim University. Okay. I'll tell you about this cash at the end of it. Yes. So the much awaited and the prestigious first award goes to Grima Jindal from Jamia Hamdard. Very pleasantly privileged to receive awards twice. I don't know. So now I announce the winners of the prize winners for oral presentation. The third prize goes to Dr. Shishad Ahmed from Jamia Hamdard. The second prize goes to Dr. Mahajan from Department of Zoology, Allahabad University. And the prestigious first prize goes to Dr. Barkha Shakia from the Department of Zoology, Aligarh Muslim University. So that's all. Congratulations to all the prize winners. Wonderful job. Well, this time unusual kind of selection has been done because of a shortage of time. Diawar Rahman and his organizing team decided that a preliminary round of presentation has already been done on the 16th, and there were juries to select the best 3 presentations for oral and poster. There were many, and many of them also came only online, and some of them were present in person. And out of those, these 3 in the oral and 3 for the poster, the competition was held today. Posters was put and oral presentations were made. And they had another set of juries, and they sat on judgment in selecting the first, second, and third. So I thank all the juries in the first instance and in the second instance for the wonderful, I want to disclose the name. So I thank the juries for the wonderful job they have done in identifying the best presenters and ranking the talents. So having said this, it's a minute for me to tell you the genesis of this award. Well, this award this year comes from a big company in France, the cell technology company, Matec. They are people who are manufacturing reconstructed human epidermis, which is a tissue model for use for skin and corrosion and skin and eye corrosion and irritation, and many other products. And a scientific advisor of the company is an associate of our society for a long time, Dr. Christian Pelvoisin. And more than the scientific association and the academic association, it's also personal also. He's so friendly with me, the president, the vice president, and all of us, and senior Pradiksha is good friendly with, I'm sorry, good Christian Pelvoisin, right? So he is friendly with all of us. And so when we announced this conference, he came forward to say that he will bear the cost of the awards this year. So the money that was given to the award winners has come from Dr. Christian Pelvoisin. Oh, he's there. So he does it. He's very much there, and so he has donated this money from his company, the Matec from France. Very, very nice of him. Very generous of him to come forward without my asking, without anybody asking. He came forward to say that this year this is my job to give you the money for the award. The award goes like 5,000 for the first each oral and poster, 3,000 for each oral and poster, and 2,000 each for oral, third prize oral and poster. So first prize 5,000, second prize 3,000, and third prize 2,000. 20,000 rupees donation from Matec company from France. And I'm so glad that Christian is there listening to and viewing what I'm saying, what I'm doing here. So I thank the organizers for this wonderful opportunity again for me to come forward to the mic and speak for a minute. Thank you so much.
Thank you, sir. Before we move forward, I would like to take a moment to hear from our audience. Your feedback is crucial to us, as it helps us to understand the impact of this conference on each of you. Please feel free to share your thoughts and insight with us. Anyone from audience? Moving on. Now we would like to welcome Dr. Irfan Ahmed Khan, sir, to deliver the vote of thanks.
Assalamu alaikum and very good evening. Ladies and gentlemen, I deem it a great honor and privilege to propose vote of thanks on behalf of the organizing committee. First of all, I thank the Almighty Allah for blessing us to make this conference a resounding success. I express my deepest gratitude and thank our chief guest and Dean Students Welfare, Aligarh Muslim University, Professor Abdul Alim sir, who despite his busy schedule has graced this occasion. Thank you, sir, for accepting our invitation and being with us here. Your words of wisdom has always encouraged us. Thank you, sir. From the depth of my heart, I thank the guest of honor for today's valedictory function, Mr. Puran Chand, Assistant Commissioner Drugs, Aligarh Division. Your presence and gentle words are a source of encouragement for all of us. Thank you, sir. I thank all the executive committee members of Society for Alternatives to Animal Experiment in India, especially the President, Professor Y.K. Gupta, sir, and the General Secretary, Professor M.A. Akbar Shah, sir. Thank you, sir, for entrusting us to organize this conference at Aligarh Muslim University. I sincerely thank the AMU administration as well as the Dean and the Principal of Jain Medical College for their unstinted support and guidance towards organizing this conference. I must mention our deep sense of appreciation to the distinguished guest speakers for accepting our invitation and for traveling from different parts of India and abroad. I am sure that the deliberation must have benefited our participants. I hope that your stay at Aligarh Muslim University must have been pleasant. I thank all the faculty members, dignitaries, residents, students for gracing this occasion. An event of this dimension cannot happen overnight. It requires meticulous planning and execution. And we are fortunate that we have the energetic and the dynamic team of the organizing committee. A special mention to our organizing chairman, Professor Muhammad Afzal sir, the dynamic organizing secretary, Professor Sayyid Ziaur Rahman sir. I must mention that we should clap at least once for him, that he has taken all the pain day and night in the organization of this conference. I thank the co-organizing chairman, Professor Farhan Ahmed Khan, and the joint organizing secretary, Dr. Jamil Ahmed sir, who have worked day and night. Dr. Jamil Ahmed sir requires a special mention because all the arrangements at the guest house was made by him, and despite all the events going on in the university, he has worked day and night so that your stay in this university must be a memorable one. Professor Sayyid Ziaur Rahman sir is hardworking and a great team leader, and he helped us with all the events without batting an eye, and for that we are so grateful. We feel privileged to work under his leadership. Thank you, sir. Special mention and my gratitude to all the teaching and the non-teaching staff members of the department, and a special mention for the young junior residents and the senior residents who have worked really day and night. They have been here in the conference for the last 2 days, sitting on the chairs without moving here and there, and that is really recommendable. I thank you everyone and for extending your support, your willingness to take this conference to another level. You have taken the responsibility very sincerely, and we are grateful for your contributions. I must congratulate all the prize winners, and I thank our collaborators. A special mention to the Jai Research Foundation, PETA, CSIR, Doran Camp Foundation, Dawakhana Tibbiya College, Aligarh Muslim University, Dr. K.C. Singhal Hospital, and last but not the least, the Ibn Sina Academy for their support for smooth organization of this conference. I also extend thanks to the staff members of Dean and the Principal Office members from the audiovisual section, and last but not the least, the non-teaching staff members of the Department of Pharmacology. Special mention for them. They are sitting here, and it's almost, they will be returning after having the dinner only. Thank you very much to all of you. I would also thank the PRO Aligarh Muslim University, Mr. Omar Salim Pirzada, and the APRO, Mr. Zishan, and the members from the media for their interest in covering this event. I thank everyone whose contribution has made this conference a successful one, and beg pardon if there were any glitches or any mistake on our part. Thank you, Dr. Anam and Dr. Abhinav, for comparing this event. Once again, I thank you all for your kind attention. Thank you.
Thank you so much, sir. Let us this be a harmonious end to a day filled with knowledge and collaboration. Thank you all for your active participation. Now I kindly request the audiovisual team to play our University Tarana, followed by the national anthem. Thank you all for being a part of this remarkable journey. Now we request you all to move to the poolside for dinner.